Welcome back to Medical Neuroscience, and welcome back to my home. It's a lovely evening as the sun is setting. It brings to mind our topic for today, and that is thinking about transitions in sleep and wakefulness. Transitions of brain and body that are tied to our daily rhythms. So today's session is going to relate to a couple of our key core concepts in the field of neuroscience, namely that neurons communicate using both electrical and chemical signals. We haven't quite given up yet on this core concept. It continues to obviously be foundational for everything we're learning about how the brain works, and today we're going to focus in on a set of neurotransmitter systems that are especially important in transitions in brain state. We're going to be talking about circuitry in the brain that is really remarkably conserved, even down to some of the molecular details as to how this circuitry operates. So we're going to be talking about circuitry that's genetically determined and is foundational for the function of the nervous system. And I hope you'll appreciate that the fundamental discoveries of these kinds of systems in the brain that can modulate brain state, sleepiness, wakefulness, these are very important concepts that are essential for understanding how to best promote health and wellness. So these kinds of fundamental discoveries are what will promote healthy living and the treatment of disease. And I think as many of us can relate to rather easily, disorders of sleep and disorders of being able to optimize one's vigilance, one's attention throughout the day. These are huge issues, and those that require a deeper understanding of the brain mechanisms involved. This topic has several learning objectives that I'd like for you to take away from this tutorial. I'd like for you to be able to discuss the concept of circadian rhythms and provide some examples of homeostatic functions and overt behaviors that are modulated on a circadian or a daily rhythm. I would like for you to be able to discuss the underlying neural systems that account for circadian rhythmicity in body and brain. I'd like for you to be able to describe the basis of brain waves, that is, the rhythms in the brain that can be detected from the surface of the scalp via means of electroencephalography. And I'd like for you to be able to use these methods to characterize the stages of sleep. And these will include stages that we call non-REM or non-REM sleep, as well as REM or REM sleep. And lastly, I want you to be able to describe the functional states of thalamocortical projection neurons. These are the neurons that project from the thalamus to the cortex. And I want you to appreciate how their state can change, and that this change in state can be used to understand the transitions from wakefulness to non-REM sleep to REM sleep, and back up through stages of sleep towards wakefulness once again. Well, I'm going to do things just a little bit differently in this tutorial. I don't have study questions for you at the end. Rather, I just want to seed our discussion with a few questions right at the beginning. I'm just going to talk you through these. They will require a yes or no answer, or perhaps a maybe of an answer, but I'm not going to implement this on the website, so there'll be nothing for you to click. I just want you to think and respond when I prompt you to do so. Okay, so we've got some questions about sleep that hopefully will set the stage for the remaining time that we have together in this tutorial. Okay, so here's the first question, well, really a statement, but we'll think about it as a question. Having a good night's sleep is essential for good health. What do you think, yes or no? I hope we all agree that the answer is a definite yes. A good night's sleep is essential for good health. prolonged sleep deprivation can be lethal. Well, indeed, this is the case. We know this from animal studies and unfortunately some instances of human torture where we suspect that part of the consequence of the mistreatment has been the impact of chronic sleep deprivation. Okay, we're going to talk some about dreams today. So what do you think? Having dreams during sleep is essential for good health. Well, this one I'd have to say is a maybe. I think that dreams are adaptive, and they certainly are pervasive throughout not just humans, but also non-human mammals seem to have something like dreams. And therefore we imagine that they must be adaptive since they're throughout our mammalian phylogenetic lineage. However, there are individuals who, because of either dysfunction or because of medication, are not able to dream. And these individuals seem to get along pretty well. I think if you were to ask them, they might say, well, their health could be better, but perhaps they have no major complaints. So this remains a bit of an open question, whether having dreams during sleep is essential for good health. But hopefully research will help address that as we go forward. Okay, have another question for you about dreams. Some people dream regularly, others don't dream regularly, and some people never dream at all. I suppose I'm talking now about people that don't have a disease or a dysfunction of their brain state system, nor am I talking about individuals that are on medications that are interfering with their capacity to dream. So with those two caveats stated, what do you think? Are there people out there who don't dream? Now, I've got members of my own family who have told me more than once that they just don't dream, or it's been months since they've dreamt. So what do you think? What's going on there? Well, I think we're going to answer that as we get further along in this tutorial, but I suppose what I would say is that these folks are dreaming, even though they are not remembering their dream. And that's probably a good thing. That's probably a healthy indication of a good night's sleep. Okay, I've got some more questions for you. Sleep is a time when the brain and the body shut down for rest and relaxation. Well, since I wrote these questions, I know that there's a bit of extremism going on here. Shut down is really too harsh a phrase. That sort of gives away the answer, perhaps. What we're going to discover is that the body and the brain, they do rest and relax, and we think that there's an important restorative function to sleep. However, the brain is far from shut down, and certainly the body is not completely shut down, otherwise we wouldn't survive the night's sleep. But there are some fascinating changes in body state that accompany changes in brain state, and we do want to talk about that for both REM sleep and non-REM sleep. All right, well, I suppose I'm giving you sort of a lecture, even though you're free to stop and start this at any time. But suppose you happen to be one of those people that doze off during lecture. I've got quite a few of them in my classes here on campus, so I know of which I'm speaking. If you are that person, do you think you might need more sleep at night? Yeah, I'd say you do. I think maybe a question is, might you even have a disorder of your sleep that requires some kind of intervention? We're going to talk about sleep disorders near the end of today's tutorial. All right, one last question. If you snore loudly at night and are excessively sleepy during the day, might you have a sleep disorder? Maybe you know some of those folks. Maybe some of those go to classes with you and are sitting next to you in lecture and snoring away. Indeed, these folks, they just might have a sleep disorder, and someone needs to get into their life a little bit and figure out what's going on. because we shouldn't consider it to be normal for people to be falling asleep and snoring excessively during the day. Okay, well, hope those questions got you in the frame of mind, perhaps the state of body and brain, to think together with me about this topic. So let's move on and begin to consider some various aspects of sleep. Okay, well, I think we're ready to move on, and we'll talk about some of the parameters of sleep and then begin to dive into some of the mechanisms. And as we get there, I do have one more question for you, and that is, how much sleep is enough? Well, this is a distribution of sleep time for a large cohort of individuals, so I think these data are fairly reliable. They seem to be fairly consistent across human cultures and across time. And it seems as though what we require is somewhere in the order of 7.5 hours of sleep. But of course, this is just population data. And I wonder, where might you be on this curve? Might you be right here at the mode of this distribution? Perhaps you're here on the rightward tail. Well, if you're like me, and I suspect that you are, you may find yourself over here somewhere, and hopefully moving in the upward direction and not slipping further down to the far left. But in any event, I think the answer to the question, how much sleep is enough, is answered by that duration of time that allows you to rise in the morning feeling refreshed and ready for the work that you have that day. And that answer, how much sleep is enough, very well may be a larger number than what characterizes your life right now. So maybe you and I together can agree that we're going to work on this and try to more optimize the amount of sleep that we get on a daily basis. Well, it turns out part of the answer to that question may depend upon where you are across the age and stage of life. And here are data that plot the normal amount of sleep from a fetus on through embryonic life into the postnatal period and all the way through older adulthood. And what we see is that just in utero, we find that our embryonic forms require the greatest amount of sleep. So here's a fetus into the last third of gestation, and we see that they're sleeping almost all the time. And this can be measured in various ways, and we'll talk about some of those ways in just a little while. But as that third trimester begins to roll towards full term, we see that there is a decrease in the amount of sleep that's required, and it continues to diminish throughout the early years of life. So you'll notice this gradual decline in the amount of sleep that people experience throughout life, and note in particular how it trails off through our older adult years. This is often a problem for older adults who want to have more of the sleep that they experienced when they were in their younger years. And changes in brain and body simply are not facilitating the regular duration and depth and quality of sleep that they may have had when they're younger. So that's one of the many accommodations to growing older that we face as we age. So this sort of begs the question then, and the question would be then, why do we sleep? Is sleep essential? Well, we know that sleep is essential from experimental studies such as this. And this is really quite a devious experiment, and I'll just take you through this. So there are two laboratory mice that are connected with a means to record their brain waves. We'll talk a lot more about that as we get further into this. And these mice are held within a chamber that's sitting on top of a turntable. Now, this turntable is triggered by high amplitude brain waves that are recorded from the experimental animal. And the experimental animal is this guy back here, this white mouse. And so whenever this mouse starts to fall asleep, we'll describe this in a little bit, there's a marked increase in the amplitude of this animal's brain waves. Well, once these brain waves hit some trigger point, they turn on a motor that causes this stage to rotate. And so what this means is that this experimental animal basically is being agitated whenever it tries to fall asleep. Now this brown mouse over here, this animal can sleep while the white mouse is active and otherwise awake. So this brown mouse, even though he's confined within a small compartment, is going to do just fine. But the white mouse, on the other hand, is in for real trouble. So this is what has happened now to this white mouse over time. We see that after a couple of weeks of sleep deprivation, things really begin to change rather dramatically for this animal. We see that despite a steady increase in food consumption, going out about 3 weeks of sleep deprivation, body weight is really collapsing on this animal. So this is just an indication that there's just widespread organ failure. And after several weeks of this experiment, this animal will die unless it's given an opportunity to acquire sleep. Now you may be wondering about human beings, and I think in your textbook you can read about what is essentially the longest known voluntary period of time where an individual has been deprived of sleep. And this went on for many, many days, and the individual then tried to catch up following the conclusion of the experiment and seemed to be okay in the end. But I suspect that there may have been some at least short-term, if not long-term consequences to this extended period of voluntary sleep deprivation. So the answer to the question, is sleep essential? Absolutely, sleep is essential for life. But why do we sleep? Well, we're going to try to answer that at least partially as we go. I suppose I can just tell you up front, the short answer is that nobody really knows why we sleep. There's general consensus that there is a kind of restorative function that takes place when we sleep. And there's even some speculation that it might actually be a conservative mechanism that is associated with endothermy, or warm-bloodedness, that allows our cells to retain a reserve in the form of chemical energy, in the currency of adenosine triphosphate, ATP. But these ideas are still really unproven, and there's a lot of deep thought, both scientific and philosophical, that has gone into addressing this question as to why do we sleep, and we don't yet quite have a definitive answer. Well, although we don't know why we sleep, one thing that's quite clear is that we should sleep every day. And sleep is one example of many rhythms in our body and brain that operate on roughly a daily cycle. These are called circadian rhythms. So they're really a diverse set of physiological and homeostatic functions that follow these kinds of daily rhythms. And the period of these rhythms is approximately 24 hours. So here's some examples. On the top we see a record of body temperature. In the middle, a measurement of the production and the circulation of growth hormone in the serum. And on the bottom, the hormone cortisol. And we see that they each show their own kind of daily rhythm. In the case of body temperature begins to fall just about the time that we begin to approach our usual bedtime. And body temperature is basically bottoming out during our sleeping time, and then it begins to rise close to the time that we were normally awake. So this is pretty good evidence that perhaps sleep has something to do with thermoregulation. Looking at various hormones, we see that some hormones seem to rise and peak as we are preparing for sleep, and then fall and remain low for the duration of the night's sleep. Other hormones begin to elevate, perhaps in preparation for the onset of activity once we awake. That might be the case for cortisol. Now, these daily or circadian cycles are actually quite closely aligned to our environmental cycle of 24 hours. But there's quite a bit of interesting neurobiology that explains how that happens. As it turns out, we have within our brain a circadian clock, a daily clock. However, the period over which that clock operates, as it turns out, is a little bit longer than 24 hours. So here's some experimental data from human volunteers who were willing to sequester themselves in a room that could be deprived of cues about day and night. And so when the cues were present, we see that these subjects were active and sleeping for a 24-hour period, and the period tended to start and stop at about the same time each day, being entrained by the presence of cues in the form of perhaps a radio signal or a television, something of that sort. But when these cues were now deprived from this environment, we see that there is a gradual shift forward in time of these daily cycles, suggesting that, at least for this cohort of subjects, the average period was longer than 24 hours, actually by quite a bit of time. We actually think that the period is not 26 hours, but much more closer to 24 hours. But nevertheless, it's longer than our environmental circadian cycle that's tied to the solar day. And in this experiment, when the cues are once again restored, now the subjects are more or less entrained to the environment going forward. So where does this kind of circadian rhythmicity come from within the brain, and how is it tied to environmental signals? Well, the answer is that there is a set of cells in the nucleus of the hypothalamus called the suprachiasmatic nucleus that contain a molecular clock. And this molecular clock is responsible for generating a circadian rhythm. Now, this molecular clock is then entrained to the environment via projections from the retina. So here we have a familiar figure for you now, I'm sure. It's a depiction of our central visual pathways. And while when we studied vision, our focus was on the projections from the retinas back to this visual thalamus, which then relays signals into the striatal cortex, right? Well, we also mentioned very briefly that there is a projection from the retina to the hypothalamus, which is focused on this small nucleus, the suprachiasmatic nucleus, which is in the hypothalamus, and it's the regulator of circadian rhythms. It's where these rhythms are actually generated. Before I tell you more about the suprachiasmatic nucleus, I just want to say a word about the retinal cells that send their projections into the anterior hypothalamus here. This projection actually comes from a population of ganglion cells that contain a photopigment. So you'll recall that when we talked about the retina, of course we emphasized the fact that our photopigments reside in photoreceptors, which are a distinct type of cell, exemplified by rods and cones, that are in the most distal part of the retina. And as you may remember, one of the surprises about phototransduction for cones and rods in the retina is that when a light turns on, these cones hyperpolarize. Remember, there's a dark current where these cells are somewhat depolarized relative to all other neurons. When they are present in the dark and when light strikes the photopigment, a transduction cascade unfolds, and the result is hyperpolarization. Well, it turns out that's not the case for the photosensitive cells that send their signal into the hypothalamus. these are a unique class of ganglion cells. We can call them photosensitive ganglion cells, and they contain a pigment called melanopsin. And you see what happens relative to the cones. When light is turned on, there's a bit of a sluggish response where the cells are very slow to depolarize, and then once they hit threshold for firing action potentials, they're ganglion cells after all, so they can fire action potentials. There's now a long barrage of action potentials, and even after the light shuts off, the action potentials and the depolarization envelope continues for some period of time. So these are cells that are not going to be very good at all for contributing to image formation in the brain. But on the other hand, they may be quite adequate for sensing the overall level of illumination in the environment and then communicating robust signals into the brain regarding slow, gradual changes in illumination. So the projections of these cells then go to the suprachiasmatic nucleus, and some really wonderful experiments were done that really helped to establish neurons in this nucleus as being the master clock for the body. For example, we know that if we can isolate these cells from the hypothalamus, basically take them out of the brain and grow them up in a culture situation, that these cells will continue to generate a rhythm that is roughly a day's length in period. Also, we know that if we take away these cells, if we ablate them from the brain of an animal, then the circadian rhythms break down. And we even know that if we put the suprachiasmatic nucleus neurons of one animal into an animal whose suprachiasmatic nucleus has been destroyed, then circadian rhythms can be restored in the recipient animal from this donor. So taken together, this is very good evidence suggesting that the suprachiasmatic nucleus harbors the cells that generate the circadian rhythm. And fairly recently, molecular biologists were able to tease apart the genes and the gene products that are responsible for creating these circadian rhythms. And it turns out there's really a beautiful set of genes and proteins that are turned on and that then feed back upon the transcription of the very genes that produce the proteins in question. And there are essentially two sets of complementary pathways that feed back upon each other. And as they do so, they generate a rhythm. So here's roughly how this goes. So as light is striking the retina and activating those photosensitive ganglion cells, action potentials arrive in the suprachiasmatic nucleus and activate these cells. And with light activation being conveyed to these cells, they turn on a set of genes. And among the gene products that are turned on with light activation are genes that produce these proteins called clock and BMAL1. So these genes turn on these proteins are produced in the cytoplasm, and these proteins then associate with one another as dimers. And then they enter the nucleus of the cell. And they form this complex that can bind to regulatory elements of target genes. And those regulatory elements are called E-boxes. And these basically form transcription factors that can then turn on a set of target genes. Well, those genes produce their products, and these include a variety of proteins. The ones we understand best are these proteins CCG and proteins from the PER family, PER3, PER2. Several of these proteins will aggregate and form their own dimers that then translocate to the nucleus. And as they do so, they inhibit or they prevent the binding of this BMAL1 and clock protein dimer to these E-boxes. So this is a form of negative feedback. And it takes some time for this to unfold. So obviously when you transcribe a gene and produce messenger RNA and then translate that to protein, there's a certain half-life to the viability of the message and then the protein product itself. So as the protein and the transcript begin to degrade, there's a self-limiting period to the duration of this cycle. So that fact, together with this negative feedback, ensures that this period is going to run for just a little bit more than 24 hours, maybe about 24 and a half hours. So it will take about that length of time for this cell to be reset and ready to again receive a signal about environmental illumination from this retinal hypothalamic pathway. Well, I think there's a much more cogent presentation of these molecular mechanisms for those of you that are interested in it, and you can find it on the website that supports our textbook, and I've provided for you a hyperlink in your tutorial notes, so hopefully if that link's working, just click on that and that will take you right to this tutorial. Alright, so far what I've told you is that these circadian rhythms can be entrained from the retina, and they generate these cycles of activity that with retinal input are tied to about a 24-hour solar day. So what then are the downstream consequences of these endogenous rhythms that are entrained to the environment? Well, one such consequence is the regulation of hormones that then can affect widespread systems throughout the brain and the rest of the body. And one important hormonal system that's activated in this way is the melatonin system, which is produced by the pineal gland. So here's a bit of an illustration showing you the pathway by which this happens. So here's our retinal ganglion cell providing input to the hypothalamus, the suprachiasmatic nucleus here, and the suprachiasmatic nucleus has projections really to a number of other hypothalamic nuclei. One of them is this paraventricular nucleus. You may remember from our consideration of the visceral motor system that the paraventricular nucleus has some large cells that grow axons all the way down to the intermediolateral cell column of the spinal cord, where these cells then can activate our preganglionic sympathetic neurons. And some of those preganglionic sympathetic neurons will synapse on the superior cervical ganglion and activate our ganglionic sympathetic neuron that innervates the pineal gland. So in this way, turning on the suprachiasmatic nucleus and the hypothalamic circuit at just the appropriate time during our circadian cycle can lead to the production of melatonin by the pineal gland. And melatonin is a very important hormone. It begins to promote a transition from wakefulness towards drowsiness and eventually sleep. So melatonin is one of the more proximal signals, not the only one, but one of the important signals that helps to promote a change in the state of body and brain in the direction towards sleep. So here's the data showing the production of melatonin throughout our circadian rhythm. And as you can see here, the production of melatonin rises in the early evening typically and continues to rise after we've fallen asleep. And then somewhere during the night's sleep, the levels of melatonin fall precipitously as we approach daybreak. And as we awake and begin our day, they remain low throughout much of the day until they start building up again in the later afternoon. So this daily cycle of melatonin is an important consideration in modulating our daily rhythms of sleepiness and wakefulness. Some of you may have tried taking a commercial version of melatonin as a way of dealing with jet lag or perhaps other kinds of disorders of sleep. And unfortunately, it doesn't seem to work that well for everyone taken exogenously. We don't really know why that is, but at least some individuals will certainly report a great benefit of melatonin as therapy for dealing with insomnia, either because of jet lag or other environmental or behavioral causes, or because of some underlying disorder of sleep. Well, okay, I think we're ready for a bit of a transition now to talk about stages of sleep and how they can be recognized clinically and how they can be monitored. So before I can describe in detail for you the stages of sleep, we need to have a bit of a primer on electroencephalography, which is a means of recording this ongoing activity of the human brain, which we sometimes call brain waves. Well, I had opportunity to think about brain waves fairly recently, as I had a very quick vacation away from recording these videos with you all, and got to go to one of my favorite places on the planet and enjoy a cool but beautiful day by the ocean, watching waves and thinking about waves in the brain. So I can't help but to watch the waves on the ocean and think of waves of activity in the brain, and wonder how turbulent are the seas of electrical activity at the moment going on in my brain, perhaps yours as well. And looking at the ocean is just an intriguing reminder of the rhythms in nature, and those rhythms are present in the patterns of activity we have in our brain. Sometimes we forget that. We consider what's going on in our brain as being totally driven by what we're seeing or what we're hearing or what we're saying, but in reality, our brain is carrying on its own rhythms, very much like these waves before me. Well, I always feel invigorated when I visit that spot, and so with some of those images in mind, let's move on and talk about waves in the brain and how we can measure them. So the electrical activity in the cerebral cortex, of course, is ongoing, and it reflects the integrated functions of really tens of billions of neurons across the cerebral hemispheres. And this activity can generate macroscopic currents that are flowing through the tissues. And these electrical currents can be recorded from the scalp on the external surface of the head. And this is the basis of this technology called electroencephalography, which is what we see here. So the scientists and clinicians have standardized the numbers and the placements of electrodes on the scalp that allow for systematic sampling of the cerebral mantle as it's present on the superficial aspects of the brain. So what we're looking at here is the placement of these electrodes as they are done in a standard experimental or clinical environment. So here's an illustration that attempts to provide a bit of the principle involved here. So imagine that a neuron is receiving input from some source, and as these neurons receive that input, they're depolarized and ions are flowing across the plasma membrane. And for excitatory postsynaptic potentials, that means positive charge is entering the postsynaptic process. So as that positive charge is entering the cell, that's essentially an inward current. So that is a current sink. There is a negativity at that site where there is this aggregate of active synapses. So positive charge is going to flow through the extracellular fluids towards that current sink. So where that current is coming from then is going to become a source of current. And so there are these sinks and sources, these current dipoles that are in register throughout the cortical mantle because of the organization of these pyramidal cells. So wherever these pyramidal cells are oriented perpendicularly to the surface of the skull, we have the potential to be able to record these electrical dipoles with the placement of an electrode close to the surface of the brain. Now obviously, as the sulci of the cerebral mantle fold in upon itself, it's going to be very difficult to record dipoles that are buried in the depth of a sulcus. So electroencephalography really is limited to these more superficial dipoles that are present on the crests of the gyri of the cerebral hemispheres. Well, what we actually record then reflects the aggregate activity of these neurons. And so at any one location, we're going to have the active inputs that are engaged in some kind of pattern of neural activity. Whether we actually see that at the surface of the scalp is going to depend upon a variety of factors. Mainly the number of active neurons that underlie the scalp electrode, the firing rate of those active neurons, and most importantly, the synchrony of the active population. So here, for example, we see a cluster of neurons receiving inputs, and when these inputs are firing in a regular fashion, we find that each one neuron might be generating electrical signals that, when attenuated in time, have the form of these various waves. And the algebraic sum of those waves, which is what would be seen with the scalp electrode, is a low amplitude but higher frequency kind of pattern. So this is consistent with an irregular pattern of activity at the level of inputs to a population of neurons. This is actually consistent with the transfer of information and the processing of signals that might be going on within a cortical column. Now, on the other hand, if it were possible to synchronize the inputs to this population of cells, we may find that many neurons are essentially engaged in firing at more or less the same time. And these signals might then sum algebraically and produce a much more noticeable wave in the electroencephalographic recording from the surface. So when there is synchronous input activity to a local region of the cerebral cortex, we tend to have higher amplitude but lower frequency oscillations that can be recorded. And as we'll see, this synchronized pattern of activity is indicative of non-REM sleep. Well, there is another context in which one can see highly synchronous activity, and it's not good. Highly synchronous electroencephalographic activity can be the hallmark of a seizure. So this is a representation of electroencephalographic data from an individual that experienced a seizure. So here we see healthy looking, high frequency, low amplitude activity recorded between pairs of scalp electrodes. And then with the onset of the seizure, we see this massive synchronous discharge as indicated by these high amplitude, lower frequency waves that emerge in this patient synchronously across the entire brain. So this would be an example of what we call a generalized seizure because it involves almost the entire brain all at once. So this pattern of brain activity would be considered to be pathological. So putting that aside for the moment, normal patterns of electroencephalographic activity generally fall into 4 recognized frequency bands. Beginning with the lowest frequency bands, we can see healthy brain oscillating in very large amplitude, very slow waves in what's called the delta band. This is 1 to 4 cycles per second, 1 to 4 hertz. This, as we'll see, is indicative of the deepest states of sleep. The next highest band of activity that is recognized is called the theta band, and this is a rhythm between 4 and 7 hertz. Theta activity is consistent with being awake and perhaps being engaged in some sort of active exploration of your surroundings. Next we have the alpha band, the alpha rhythms. Alpha is between 8 and about 13 hertz, and the alpha rhythms seem to be a prominent feature of our sensory cortical areas, especially when those cortical areas are somewhat disengaged from the sensory environment. So for example, if one were to close one's eyes, you'd find that the visual cortex very quickly will go into a alpha rhythm with large amplitude waves in this 8 to 13 or so hertz range. And when we open our eyes, for most people anyway, the alpha rhythm will go away just about immediately. So the alpha rhythm is an indication that a sensory cortex is in a state of quiet rest. Now the highest frequency waves that we typically recognize are in the beta range. Sometimes we split this into beta and gamma, with beta being anything above 12 hertz basically, and gamma being around 40 to 60 hertz. So gamma waves, beta waves, these are high frequency but very low amplitude waves. So this would be indicative of a high degree of irregular activity or high degree of desynchronization within cortical networks. And this might be consistent with the fine granular processing of information that is happening at the columnar or perhaps the modular level within our cortical networks. So electroencephalographic recording is very important from a clinical perspective. It can be used to diagnose disorders like epilepsy or seizure disorder, and it can be used to investigate various stages of consciousness, including the normal transitions from sleep to wakefulness and then back to sleep again, as well as brain states that are associated with injury to the brain itself, including coma and minimally conscious states of activity. So let's now see how electroencephalographic data can be used to characterize the various stages of sleep. Well, the electroencephalogram has provided psychiatrists and other kinds of physicians who study sleep the opportunity to characterize the progression of an individual from being fully awake to being deeply asleep. And we use these electroencephalographic criteria to recognize essentially 5 stages of sleep, 4 stages of non-REM sleep, and then we can recognize REM sleep based on the behavior of the individual and the electroencephalographic record. So stage 1 sleep is a time when we are beginning to feel drowsy. We are otherwise still awake, but beginning to experience that transition from being vigilant to being quite drowsy. So during stage 1 sleep, we begin to see the emergence of some higher amplitude rhythms, still fairly fast, and the increase in amplitude only modest, but it's consistent with the increase in synchronicity that is going to lead to more deeper stages of sleep. So in stage 2 sleep then, this is the next deeper stage, we see an increase in amplitude, and then we see the emergence of a very particular feature that is characteristic of this stage of sleep. It's called the sleep spindle. And this sleep spindle is a very brief, just a few seconds of high frequency activity that occurs in these clusters. So it's a high amplitude, high frequency burst that is indicative of there being a great deal of synchrony imposed upon thalamocortical neurons, but only for these little brief epochs of time. And so this is something that's happening as we begin to enter our deeper stages of sleep. So stage 2 sleep might pass after 15 or 20 minutes or so, and we begin to get into a stage of sleep that more clearly is increasing the amplitude of some of these brain waves. So the electroencephalographic evidence is suggesting that the brain is getting even more hypersynchronous, but that the rhythms are also slowing. So with stage 3 sleep, we're getting into fairly moderate levels of sleep. The sleep spindles have gone away now, and the electroencephalogram is characterized by the emergence of these larger amplitude waves. But we see the deepest stage of sleep in stage 4, and this is when the electroencephalogram reveals large amplitude, slow delta waves. And these waves are indicative of a cerebral cortex that's dissociated from its environment and is engaged in this hypersynchronous slow rhythm of thalamocortical oscillation. Now, once we cycle down to stage 4, to deep sleep, after some period of time, maybe an hour and a half or so, something fairly dramatic happens. We begin to step out of that deep, slow wave sleep, and we enter a stage of sleep where the electroencephalogram reverts to a low amplitude, high frequency pattern, something that resembles actually what the electroencephalogram looks like in the awake state. This is called REM sleep. REM stands for rapid eye movement sleep. It's also called paradoxical sleep. And the paradox is, how can we be still asleep and yet have electroencephalographic activity that resembles an awake brain? Well, REM sleep is really a fascinating state of body and brain. And let me tell you a little bit about what's going on there. So, yes, it's rapid eye movement sleep, but it's so much more than that. So the cerebral cortex is in this desynchronized state, and electroencephalographically that is high frequency, low amplitude brain waves. But when we look at the brain with imaging methods, we know that there are some really interesting features of blood flow that suggests that certain parts of the brain actually have increased activity over resting conditions, whereas others have decreased activity. Specifically, those areas that are increased include structures that are in the medial parts of the forebrain, structures associated with the limbic forebrain, such as the anterior cingulate cortex, our medial temporal lobe memory system in the parahippocampal gyrus. Just in front of that, we have the amygdala. So, as we'll see in a different tutorial, these are all structures that are intimately associated with the experience and expression of emotion. And the hyperactivity of these structures during REM sleep very well may be responsible for the emotionality that seems to be present in so many of our dreams. Meanwhile, those parts of our brain that are otherwise involved in executive control functions, such as this dorsal lateral prefrontal cortex, these regions actually show decreased activity relative to the resting state. And perhaps with a decrease in the moderating influence of this executive control network, this might help explain why sometimes the content of our dreams can be socially inappropriate. So, yes, indeed, REM sleep is the phase of sleep where most, but not all of our dreams occur. I'll have more to say about dreams in just a minute. So as we cycle through our stages of non-REM sleep and then enter REM sleep, it's not just the brain state that's being modulated, but also the state of the entire body. So this can be assessed in a variety of means, and here are some data that are fairly typical in terms of what might be gathered during a sleep study if one were to go to a clinic that's specialized in the treatment of sleep disorder. Now, these are somewhat idealized data, so most of us don't have such beautiful looking cycles of non-REM to REM sleep throughout our typical night. But nevertheless, this shows a typical night's sleep where we cycle through non-REM and REM sleep. We tend to spend increasingly more time in REM sleep as our night progresses, and then less time in stage 4 sleep, and eventually even less in stage 3 sleep as we approach waking time, wherever that happens to be for you. Now, during these phases of non-REM and REM sleep, movements of our body's muscles, including those visceral muscles that are necessary to sustain life through sleep, are being modulated. So here what we have is a record of the electrooculogram, which is indicative of when we are moving our eyes. And sure enough, as we enter rapid eye movement sleep, that's where we find an increase in this electrooculogram activity. indicating rapid movements of the eyes during REM sleep. Meanwhile, this is EMG, or electromyographic data, recorded from a limb muscle. And what we see is that, of course, when we're awake, there's a fair amount of activity. And as we're falling asleep, this activity begins to subside. It begins to pick up again as we pass back through our stages of non-REM sleep towards REM. But during REM sleep, and this is especially obvious as we have longer bouts of REM sleep, there seems to be a suppression of muscular activity during REM sleep, and this is really amazing. It in fact is the case. So for most of our large skeletal muscles, there is active suppression of muscle activity during REM sleep. Now, some have suggested that this is adaptive. This is so that we don't act out our dreams with our musculoskeletal system. That may be the case. We don't exactly know how to interpret this phenomenon, but there is some really fascinating neuroscience that explains the inhibition of alpha motor neurons that innervate these large skeletal muscles during REM sleep. I'm wondering if you can hear that. The barred owls in the woods behind me are starting to go crazy over something. I don't know exactly what's going on there, but hopefully they'll calm down. Now notice what's going on in our visceral motor system. So while we are phasing into our deeper states of non-REM sleep, our heart rhythms are subsiding, so are our respirations. But as we begin to cycle towards that paradoxical sleep, that REM sleep, there's an increase in both heart rate and our breathing rhythms. And so again, this is perhaps consistent with activation of the limbic forebrain, consistent with a heightened sense of emotionality. And then also for men, men experience penile erection during REM sleep, and women experience an increase in vaginal lubrication during sleep. Again, perhaps consistent with an increase in activity coming out of the limbic forebrain, which is also highly engaged in various kinds of sexual behaviors as well. So just a couple of other interesting points about REM sleep that are worth making. While sleep is necessary for life, it appears as though REM sleep might not be necessary. As I mentioned earlier, when we were thinking together through some of those opening questions, there are individuals that are taking medications that suppress the experience of REM sleep. That would be a side effect of their medication. And these individuals seem to be getting along quite well. Their lives are not in jeopardy, at least not because of the cessation of REM sleep. Now, it may not be so clear why we have REM sleep, but most of our dreams happen during REM sleep, and so perhaps that's providing us some insight that maybe what's really important about REM sleep is the opportunity to dream. This is an idea that's been with us for some time, and it's regaining traction with some new neurobiological studies that are being done to try to explore the possible benefits of dreaming. So while REM per se is still quite mysterious in its significance biologically speaking, perhaps it's at least providing a space for the experience of dreaming, which we'll turn to in just a moment. So, as Hamlet said in his famous "To be or not to be" soliloquy, to sleep, perchance to dream. So, many of us, like Hamlet, have wondered about dreaming and what it might actually be doing for us. And, of course, many over the last 150 years or so have argued vigorously about this. Certainly, Sigmund Freud had his ideas about the importance of dreams. Freud argued that dreams provided a means for the ego to relax its grip on the id. Now, most of us don't really adhere to these Freudian concepts. We want to replace them with neurobiological concepts. And so we're not quite sure what to do with this one, but there may be a sense in which Freud was ahead of his time, at least anticipating some of the brain mechanisms that reflect a difference between explicit processing in the brain and implicit processing. Well, there's an idea that says that dreams are really an opportunity to rehearse some of the less common behaviors that are not often expressed in the waking state. For example, very intense emotional states, perhaps aggression. So various kinds of behaviors that might be risky and self-destructive if they were to occur during wakefulness might be played out vicariously through dreams. Well, there's another idea. It says that a dream is an opportunity to unlearn, that is, to erase some kind of non-adaptive pattern of neural activity, perhaps stored as a memory. So this could be a very useful mechanism for us to get rid of unwanted memories. Well, there's another thought that seems to be gathering a lot of interest these days, which is exactly contrary to this last one. Rather than dreaming being about unlearning, the prevailing view, I think, among neuroscientists today is that dreaming, in fact, provides an opportunity for synaptic plasticity. That dreaming provides us with opportunity to consolidate memories, to engage mechanisms of synaptic plasticity, and retain the learning of the day. So there are a variety of studies that are showing evidence for this view, from behavioral studies to neurophysiological studies, and it's quite exciting to see this emerge as potentially a neurobiological mechanism that explains the benefit of dream. Now, there's so much yet to be discovered about this. We are far from understanding exactly what's going on here, but it's an intriguing concept that dreaming provides us an opportunity to actually consolidate the learning of the day. Well, there are still some nihilists out there that would say that dreaming is nothing but an epiphenomenon, that perhaps actually REM sleep is important after all. We just don't understand why it's important yet. But dreams per se may just be epiphenomenal. Well, I think the sum of all this is that no one really knows why we dream or even how we dream. But we are developing some concepts that I think maybe are leading us down a fruitful path. So let's see where this goes over the next decade or so, and hopefully we'll have much greater insight into both the nature of dreams as well as the neurobiological mechanisms underlying dreams. Well, it's to some of those mechanisms that we do understand that I want to turn to now. So let's consider some of the neural circuits that are important in governing sleep. So, as should be clear to you by now, sleep involves a series of transitions from one state of body and brain to another. And this reflects a complex interplay among a variety of neurochemical systems that are present in the hypothalamus, in the reticular formation of the brainstem, and these are systems that have widespread projections that can affect circuitry really throughout the central nervous system, even down into the spinal cord. But they are especially focused on modulating the way the thalamus interacts with the cerebral cortex. So I'm going to give you a bit of a detailed run through these mechanisms, and then we'll come back and talk about them in slightly broader brushstrokes. So what I'd like to do is go through this table and lay out for you some of the details about which neurochemical systems are involved in which brain states, and then we'll come back and talk about how they interact with one another. So first, let's consider the maintenance of wakefulness. So wakefulness is a product of the activation of quite a large number of neurochemical systems. Among the most important are the first two listed here in this chart, the cholinergic nuclei that are found near the junction of the pons and the midbrain, and the locus coeruleus. The locus coeruleus is a collection of cells in the dorsal tegmentum of the pons that produce the neurotransmitter norepinephrine. So together, these two, acetylcholine and norepinephrine, very important neurotransmitters in maintaining wakefulness and maintaining vigilance. But in addition to acetylcholine and norepinephrine, we also have other neurotransmitters. The serotonin system, which are derived from the midline raphe nuclei that are found right in the seam of the brainstem. Raphe meaning seam. There's an important nucleus in the posterior hypothalamus called the tuberomammillary nucleus. This nucleus produces histamine, and histamine, when it's active, promotes wakefulness. The lateral hypothalamus is home to collections of cells that produce a variety of peptide neurotransmitters. One of them is this peptide called orexin, sometimes called hypocretin. And orexin, when active, promotes wakefulness mainly by stimulating these tuberohypothalamic cells that then release histamine. But orexin has other roles to play in other circuits that project out of this lateral hypothalamus. So these are systems that promote wakefulness. The systems that then promote falling asleep, feeling drowsy, involve basically a decrease in the activity of many of the ones that I've just mentioned. So the functions of acetylcholine, of norepinephrine, of serotonin, they are all decreasing as we are entering the stages of non-REM sleep. So we're going to talk a little bit about the neurochemistry as to what's pushing these systems that were active in the waking state towards decreased activity in non-REM sleep. Well, this leaves us then with a consideration of the neurochemistry of REM sleep. Now, in REM sleep, we have a bit of a hybrid state where the brain is active, the body is in this suppressed, partially paralyzed condition. And this dichotomy between an active brain but an inactive body and yet being asleep implies that there must be some differentiation of neurochemical systems, some of which might be turned on, some of which might be turned off. And indeed that seems to be the case. The cholinergic nuclei of this junctional region between the pons and the midbrain are active, in fact, in a quite unusual pattern of activity that produces a very discrete type of electrical signature that propagates from the pons into the lateral geniculate nucleus of the thalamus and then into the occipital cortex. These are called pontogeniculo-occipital waves, or PGO waves. So there's a very distinct pattern of activity that's present in these brainstem cholinergic neurons. Meanwhile, some of the other amine neurotransmitter systems that otherwise might support a more wakeful condition are inactive. This would include our norepinephrine system and then the indolamine system involving the neurotransmitter serotonin. So both serotonin and norepinephrine are depressed at the same time that the cholinergic system is turned on in this very peculiar way. So there are a variety of mechanisms then that are operating, some of which are being pushed up, some of which are being pushed down, that help explain these very peculiar transitions that we have in our state of body and brain. Well, I'll give you a bit of an anatomical view of some of these systems now. So our cholinergic nuclei that are so important in maintaining wakefulness and then are turned on during REM sleep are found here in the rostral pons and in the caudal midbrain. And these neurons have projections that are directed up towards the thalamus. They also have some projections down towards other parts of the reticular formation in the medulla. Our raphe nuclei are found along the midline of the brainstem, from the midbrain all the way down into the medulla. And these raphe nuclei give rise to very long projections that basically extend all over the entire brain and even down into the spinal cord, where they release serotonin. The tuberomammillary nucleus is a very small nucleus in the posterior part of the hypothalamus, and it gives rise to projections that extend throughout the entire central nervous system. And these projections release histamine. And This histamine is an important neurotransmitter in supporting the wakeful, active brain. And as many of you who have seasonal allergies know, if you take an antihistamine, you are very likely to feel drowsy. And it's because you are antagonizing this natural, wakeful, promoting chemical in the brain. And finally, we have this beautiful structure found along the dorsal tegmentum of the pons called the locus coeruleus. And this is the location of the cell bodies that produce norepinephrine. And norepinephrine is right up there with acetylcholine and histamine as being among the important neurotransmitters that support wakefulness. These noradrenergic fibers extend throughout the central nervous system, releasing norepinephrine and modulating our level of arousal, vigilance, and attention. There's a couple of other key players here that I want to localize for you. If we look into the medial part of the hypothalamus, we can identify some of these clusters of cells. Here's our suprachiasmatic nucleus sitting right above the optic chiasm, of course. And back in the posterior part of the hypothalamus is where we find that tuberomammillary nucleus, just dorsal to the mammillary body. Now, our orexin neurons are those that are producing this peptide that are involved in promoting the wakeful state, and the orexin neurons are found in the lateral part of the hypothalamus, so just a little bit deep to the wall there of the third ventricle. Now, I want to also talk about this nucleus here, the ventrolateral preoptic nucleus. So this is a nucleus that's found in the anterior part of the hypothalamus, and it's very important because it seems to interact with structures in the posterior part of the hypothalamus, specifically this tuberomammillary nucleus. And the ventrolateral preoptic area, or preoptic nucleus, has an antagonistic effect with respect to these orexin neurons. So this preoptic area is going to try to suppress the activity of these histaminergic neurons among some of our other activating neurons. So there's a bit of a push and a pull that's happening here, with the orexin neurons providing the push signal, promoting wakefulness, and this preoptic region providing the pull signal, which is trying to pull the brain into a state consistent with drowsiness and eventually falling asleep. So schematically then, here's what this might look like. So here's our orexin neurons that are sending excitatory signals, activating our brainstem reticular formation neurons that are supporting wakefulness, as well as sending projections to the posterior hypothalamus where they're activating those histaminergic projections from the tuberomammillary nucleus supporting the wakeful state. Now, these circuits are being antagonized by this anterior part of the hypothalamus, this ventrolateral preoptic nucleus, which is suppressing the activity of this tuberomammillary nucleus, and likewise sending signals that have a suppressive influence over our cholinergic neurons and our adrenergic neurons and our serotonergic neurons of the brainstem. So this transition then between being vigilant, being awake, being attentive, and being a bit sleepy and being a bit drowsy is likely playing out in the posterior hypothalamus, depending upon the balance of these influences, whether the sleep-promoting system is winning or is the vigilance-promoting system. Now, before I say a little bit more about this, I do want to share with you an image that was taken by a student of mine just a few months ago in the brain anatomy lab. And it's a very unusual look at this beautiful structure right here, the locus coeruleus. So thanks to Jonathan Weinhold, who took this picture. what I had a student do, actually not Jonathan, but another student. We were dissecting some human brains as a way of learning human neuroanatomy. And I asked the student to make a mid-sagittal cut with a large knife through this brain. And the student was just a bit off the midline, but that turned out to be really fortuitous, because look at what we found when we looked into the brainstem following this parasagittal section. We found this beautiful band of dark substance in the dorsolateral tegmentum of the pons. And what we're looking at here is a trail of pigment that is present in the cells that produce norepinephrine. So this is in fact our locus ceruleus, which is a Latin term meaning blue spot. So this is the blue spot in the brain, or this is the dark substance. We also have some dark substance here on the medial edge of the ventral tegmental area, substantia nigra pars compacta complex in the midbrain. So we're actually seeing the dark substance in two places here in this parasagittal preparation. So thank you to Jonathan for sharing this picture with us, and perhaps you'll have opportunity one day to see the blue spot for yourself in the human brain. So let's get back to talking about this balance between the sleep promoting systems and the wakefulness promoting systems. So one of the key factors in promoting a transition from being awake to being asleep is the activation of this ventrolateral preoptic area of the hypothalamus. It turns out that one substance that is activating this region is the substance adenosine. And sleep researchers tend to be really excited these days about finally beginning to understand how adenosine seems to be operating in promoting a transition towards sleep. Of course, as you know, adenosine is the A in ATP, adenosine triphosphate, which is the main molecule that carries energy in our brain cells. So as that phosphate group is cleaved, energy is released, and that energy can be used to support a whole host of metabolic function within brain cells. Well, as it turns out, we think, as our brain cells are active during the day and energy is being consumed at a higher rate, then ATP is being broken down into adenosine diphosphate and eventually adenosine monophosphate, and then finally free adenosine is available and is released from cells and begins to accumulate in the extracellular spaces. Well, in the region just in front of the hypothalamus called the basal forebrain, there are nuclei there that seem to be sensitive to these rising levels of adenosine. So as adenosine rises in this region of the basal forebrain, it drives activity that is exciting these cells of the ventrolateral preoptic area. So the basal forebrain then seems to play an important role in driving this sleep-promoting system that's coming out of the anterior hypothalamus, leading to a suppression of the posterior hypothalamus as well as these brainstem nuclei that are otherwise responsible for maintaining alertness in the conscious state. And adenosine has been conceptualized as being a thermostat of sorts that is sensitive to the energy that's being consumed by neurons. So as energy is being depleted by these brain cells, adenosine is accumulating, signaling the body to drift off into a state of quiescence, that is, to falling asleep. And as we progress through a typical night's sleep, adenosine levels fall again. So this has led to the hypothesis that adenosine is a kind of energy thermostat in the basal forebrain region that is indicating the expenditure of energy over a very long time scale, the time scale of the daily rhythm. And this accumulating extracellular adenosine then might be one of the proximal factors in promoting a transition from wakefulness towards sleep. Now, it's quite interesting that, of course, human cultures for thousands of years have discovered that xanthines like caffeine and theophylline can block the activity of adenosine on this basal forebrain region. Adenosine binds to adenosine receptors, and caffeine and theophylline block these receptors. So while adenosine is accumulating, the impact of that accumulating adenosine on the basal forebrain is mitigated by the consumption of these caffeinated substances. So those of us that find ourselves quite addicted to caffeine are trying to fight our energy thermostat, and I suspect in the long run that's probably not a very wise lifestyle choice. So perhaps a little moderation is in order here, probably is in my lifestyle for certain. Okay, well, let's now turn to a bit of a closer look at the rhythms that are generated at the neuronal level that are reflected at the macroscopic level in our electroencephalographic depiction of brain waves. So these rhythms reflect a bistable condition of our thalamocortical projection neurons. So these thalamocortical projection neurons basically exist in one of two physiological states. They may be at times in an oscillatory state, or they may be tonically active. And as it turns out, this oscillatory state is the state that we find these cells in as we transition to deeper and deeper levels of non-REM sleep. The active state of body and brain is when these cells are then depolarized out of this oscillatory mode and are in a more tonic active state where they can faithfully transmit information from their input sources through their thalamic relays to the cortical regions that they innervate. Here's a look at these neurons in these two states. So when an animal is asleep and we look at the recordings of membrane potential from a thalamocortical cell, we see that these cells are oscillating in a fairly slow rhythm. There is a slow depolarization envelope that hits threshold and then fires a series of action potentials. And then that leads to a deep hyperpolarization. So this is the oscillatory state. The membrane potential is hyperpolarized, and that hyperpolarization triggers a slow depolarizing envelope that is attributable to the activation of a calcium ion channel. And so this depolarizing envelope is actually a calcium spike, although it's not particularly spiky because it has such a slow time course. But as this depolarizing envelope proceeds, eventually a threshold is reached for the activation of our much faster voltage-gated sodium and potassium channels. So when that threshold is reached, Hodgkin-Huxley style action potentials fire, and that leads to a barrage of activity, a little burst of activity that rides on the top of that depolarizing envelope. And after that brief barrage of action potentials, then the membrane potential hyperpolarizes again, beginning yet another cycle. So this oscillatory mode is what explains this slow wave hypersynchronous behavior that can be observed in thalamocortical systems while we fall into deeper levels of sleep. Now this hyperpolarized state is induced by a withdrawal of these brainstem inputs that are otherwise promoting depolarization. So a withdrawal of cholinergic and adrenergic input to these thalamic neurons causes them to slip into this hyperpolarized state where this burst generating mode begins to take hold of the membrane potential of these cells. And when these cells are oscillating, they are not responding to the inputs. that they might otherwise convey onto the cerebral cortex. So this oscillatory mode is a means of physiologically disconnecting the thalamus from the outside world. And we think this is quite important because it helps us to not be distracted, not be awakened, not be aroused by stimuli that fail to reach some kind of threshold of activation. So obviously that would be consistent with maintaining a sleeping condition, even with some lower level degree of stimulus in the environment. Now, suppose there is a very strong stimulus in the environment. That may be enough to sufficiently drive afferent activity in these thalamic neurons to depolarize them out of this oscillatory mode. Or it might be sufficient to activate the brainstem systems that promote the waking state. Through one means or another, depolarizing these thalamocortical neurons will shift them out of their oscillatory mode into a tonic firing mode. And we see that here with this gradual depolarization of the membrane potential. And in this instance, it was induced by activating brainstem inputs from the cholinergic nuclei and from the locus ceruleus. And after these neurons have been depolarized, now they can respond to sensory stimuli. So this now is a barrage of action potentials that is driven by the presentation of a sensory stimulus to this sensory thalamocortical neuron. So the brainstem activating systems associated with the cholinergic and the adrenergic populations are those that are really critical for inducing this state change in these thalamocortical neurons from this oscillatory mode to this tonic firing mode. The oscillatory mode is a mode where the thalamus is disconnected from the outside world, and the tonic firing mode is when the thalamus is now engaged and ready to faithfully transmit signals that it receives through the ascending sensory pathways that are running through the thalamus. Now, here again we confront the paradox of REM sleep. So the thalamocortical neurons are in this depolarized tonic firing mode state during REM sleep, just as they are during wakefulness. And this is presumably one reason why there are cogent patterns of activity in our thalamocortical networks that might be consistent with having a dream, for example. So while the information is not being derived from outside of the brain, information is being resurrected from within the circuits that have been modified by previous experience. And we have these hallucination-like experiences that we call dreams. Now you may be wondering why it is that sometimes you have really vivid dreams and sometimes you might think that you're not dreaming at all. Well, we've already made the point that we think we all have dreams every night since we have cycles of REM sleep every night, unless there should be a disorder of sleep present. And the reason why you may not remember a dream on any one given night's sleep may simply be that you didn't wake up while that dream was happening. If you do find yourself going through a period in your life where you are recalling many vivid dreams, that's probably an indication that you're not getting very good sleep and that you are waking up repeatedly during these bouts of REM sleep. So while it's quite normal to remember a dream, I think if you find yourself doing that more and more frequently, then that might be a sign that your sleep is being more disrupted than it ought to be. Speaking of sleep disorders, let's conclude our discussion of sleep by considering just a few common disorders of sleep, beginning with probably the most common one of all, insomnia. So insomnia refers to impairments associated with the inability to either fall asleep, to stay asleep, or to experience the normal restoration of energy and vigor that sleep ought to provide. So insomnia, we think, affects about 15% of the population, and it can come from a variety of sources. One fairly common cause of insomnia is simply ingesting too much caffeine. So relative to a control subject that is showing these nice cycles down through our non-REM stages of sleep, back up to REM, and then back down to stage 4, stage 3 sleep through the night, this individual who has consumed an excessive amount of caffeine is only hitting slow wave sleep for very brief periods of time, and only in the first couple of hours of sleep. So notice that the overall duration of sleep in this subject is quite short, just a few hours, compared to a normal 8 or 9 hours of sleep in a night. And the amount of time that's spent in stage 4 sleep is far from normal with this overly caffeinated individual. So again, what this person has done is they've blocked their adenosine receptors, which presumably is not activating the ventrolateral preoptic area sufficiently to counteract the stimulating systems that are otherwise trying to promote wakefulness. Now also shown here is benzodiazepine, which is a form of sleeping pill, and what benzodiazepines do is facilitate currents that are flowing through those GABA-A receptors that are being activated by endogenous GABA. And notice that with a benzodiazepine on board, one reaches stage 4 sleep much more quickly and then has much more extended periods of this deep stage 4 slow wave sleep compared to control subjects. So insomnia can be treated in various ways with pharmaceutical measures or with various behavioral measures, but it's one of the most common disorders of sleep that we have in our population. Another disorder of sleep is when we misalign ourselves with our circadian rhythms. So there's a condition called circadian rhythm disorder, and this can happen for a variety of reasons. There can be physiological causes for circadian rhythm disorder. There can be lifestyle choices, such as shift work, or simply choosing to stay up very late at night and not get a proper night's rest on a regular basis that can result in a drift in one's circadian alignment to the entrainment that our environment is trying to provide for us. Well, here I am speaking to you at night with electrical lights illuminating my environment when perhaps I ought to be drifting off to sleep right about now. So there are various ways in which we manipulate our environments and change our lifestyles that run the risk of upsetting the alignment that we ought to have with our environment. Well, another relatively common disorder of sleep is called sleep apnea. So sleep apnea results in a cessation of breathing during the sleep. Now, this can be quite a serious problem, obviously. And so this typically results from a mechanical obstruction from the airway. So people that are somewhat obese are at risk for this as our muscle tone begins to relax as we're entering our deeper stages of sleep, especially if we have some excessive weight around the region of our neck. The region of our pharynx can actually collapse down upon our airway and occlude the flow of air down into our trachea. And once that happens, then obviously we're not breathing normally, and carbon dioxide builds up very quickly and can induce a reflexive, forced inspiration of air. And that act can be very disruptive. It can wake us out of sleep. And so what we see in this slide here is a record of someone who is attempting to sleep with sleep apnea. And we can see that they never enter the deeper stages of sleep at all. They are simply bouncing around between being awake and being in the lightest stages of sleep, and having quite a fitful night of it. So you can imagine why such individuals really struggle so much in their waking state, fighting off drowsiness and the tendency to actually fall asleep during times when it would be inappropriate to do so, because they're not getting a proper night's rest when they ought to be in a situation of cycling down into those deeper stages of sleep that this condition is denying them. So again, thankfully there are treatments for sleep apnea. The most common apnea is a positive pressure system, which is designed to keep that pharynx from collapsing down upon the airway during the progression into deeper stages of sleep. The next sleep disorder I'd like to mention is called narcolepsy. Now, narcolepsy is sometimes called a REM sleep attack or sleep attack. What this is a condition that allows for the intrusion into the waking state of fragments of the state of body and brain that we normally associate with REM sleep. So typically, people who are having these sleep attacks, they're not actually asleep, but they're having elements of REM sleep intrude into their conscious state. So this can be a terrible condition to have to live with, because the full-blown experience of REM sleep is seemingly right at the threshold at any moment. So, for example, one common condition associated with these attacks is cataplexy. And cataplexy refers to a partial or perhaps even a complete paralysis, consistent with the paralysis of large skeletal muscles that we see during normal REM sleep. So imagine these attacks intruding at any moment during your normal conscious state. This is what people with narcolepsy have to experience. And in a rather cruel twist, in the experience of an individual suffering from this condition, these attacks are often brought on by emotional experiences. Intense emotions, often very intense positive emotions, such as a laughter at a pleasant and unexpected experience of some sort. That might be sufficient to induce one of these attacks. Now, what's going on here, we think, is a mutation in either the gene that produces this important peptide, orexin, or the receptor for this peptide. One way or another, it seems as though in people who have narcolepsy, there is a mutation that is affecting the function of the orexin hypercretin system. And this is going to interfere either with the ability to sustain the awake state, or it's going to interfere with the regulation of those systems that are promoting the onset of REM sleep. Now, we know so much about narcolepsy because of the discovery of some animal models that, like human patients who have narcolepsy, have mutations in the genes for these critical components of this orexin system. So thanks to these animal models and the research that's going on with them, we are beginning to understand much better how to approach the intervention and the care of these people that have what potentially could be a life-threatening sleep disorder. And then lastly, I'd like to just mention restless leg syndrome. This is a sleep disorder characterized by a feeling in the extremities, particularly the legs, that movement is necessary in order to relieve a sense of unease. This might feel like an itch, it could feel like a tingling sensation. Often it's quite hard to describe, but it's a sensation that drives a person to want to move their legs in order to achieve some measure of relief. Well, restless leg syndrome has been associated with a deficiency in iron within the dopamine neurons of the substantia nigra pars compacta. And this has suggested that perhaps there's some deficiency in the dopaminergic system, presumably projecting to the putamen, that component of the striatum that is associated with the loops that govern the movements of the arms and the legs. So perhaps there is some disorder that is manifest at the level of basal ganglia circuitry that is providing for this urge or this impulse to move the legs to achieve some resolution to these feelings. So, as I think this tutorial has made clear, sleep is a very complicated business. There are a number of neurochemicals that are involved in regulating the transitions of states of body and brain. We're beginning to learn more about them, and there are lots of opportunities for things to go wrong, either because of disease or disorder, or now more commonly because of pharmaceutical agents and dietary factors that we take into our bodies that interfere with the normal operation of these systems. So if you are in the healthcare professions, you should know quite well how the pharmaceutical agents that you may be prescribing or interacting with your patients are affecting their sleep cycles. And I hope this tutorial has emphasized for you how critically important a good night's rest is for promoting health and wellness. And we need to be all the more vigilant to protect this important aspect of what it means to maintain our life on a healthy trajectory, when there are so many opportunities for us to interfere with this process. Everything from a morning cup of coffee to the lights that illuminate our environments in the nighttime. Well, I think I've probably said enough about that subject, and after all, it is getting on into the evening, and I'm feeling my adenosine levels rising and my orexin levels lowering, so I know it's about time for me to turn in. So I'll see you next time when we will dive into the limbic forebrain and talk about those brain systems that are engaged in the experience, in the expression of emotion. So I'll see you next time, and otherwise, I hope we all have a good night's rest. Welcome back to Medical Neuroscience, and in this tutorial I'd like to give you an overview of the limbic forebrain. The core concept that will be germane for today's discussion is simply that the brain is the body's most complex organ. And I have two learning objectives for you today. First, I'd like for you to be able to review again the anatomy of the internal structures of the forebrain, with a particular focus on localizing the amygdala and the hippocampus in the medial part of the temporal lobe. So, as we'll see, this tutorial will revisit the Brain Anatomy Lab with a focus on looking at slabs that have been cut through the forebrain to reveal the location of the amygdala and the hippocampus in the medial temporal lobe. So I want you to pay special attention to where you would find these structures, and I'd love for you to have a general sense of what these structures actually look like when it is possible to cut into a human brain. My second objective for you is that I want you to be able to discuss the major functions of the limbic forebrain. Now, much of the content that pertains to this second learning objective is found in the tutorial notes that I've given you. It's one of those notes that's available in PDF format in narrative style rather than outline style. But you'll also see that in a different tutorial here in Unit 6, I'm talking to you more generally about the neurobiology of emotion. And since much of the limbic forebrain pertains to the brain systems that are involved in emotion and in related aspects of cognition, these topics will be elaborated in more depth in that particular tutorial. So let this experience here today remind you of the anatomy of the medial temporal lobe, and study the tutorial notes, and then conclude Unit 6 by considering the neurobiology of emotion, and in particular how the brain's systems for emotion and reward can be thwarted by addiction. And that will be the conclusion of Unit 6, and indeed the final tutorial presentation in medical neuroscience. So, I'll see you in the lab, and hope you enjoy reviewing again the internal anatomy of the medial temporal lobe. The purpose of this lesson is to highlight the relative position of two very fascinating structures that are found in the anterior and medial part of the temporal lobe, the amygdala and the hippocampus. And we're looking at two coronal slabs that are adjacent to one another. The slab on the left is anterior to the slab on the right. So let's first look at the slab that's on the left. And let's focus down into the anterior and medial part of the temporal lobe. So just below the basal ganglia, we see a large mass of gray matter accumulating. This is the amygdala. So here's the amygdala in the right hemisphere, and just across the midline, we can see the amygdala in the left hemisphere. The amygdala is another one of those great anatomical terms. It means almond, not because of its shape, but because the amygdala is buried deep in the temporal lobe like the meat of an almond within its shell. So at this level, we have the amygdala in the anterior medial temporal lobe. Now if we can look at the adjacent slab in the same anterior and medial regions of the temporal lobe, what we see is not an almond shape, but rather this curved bit of cortex that folds in upon itself. This is, again, the hippocampus. And at this level, the hippocampus is a broad structure that forms the floor of the lateral ventricle as it extends into the temporal lobe. So here we have the right hippocampus, and across the midline of the section is the beginning of the left hippocampus. So for the most part, the amygdala sits anterior to the hippocampus. But if we look carefully at this left temporal lobe, we can see that there is a bit of overlap. This is some of the posterior amygdala that sits just above, meaning just dorsal, to the hippocampus. So there is a plane of section where we see both the posterior part of the amygdala and the anterior part of the hippocampus. But much of the body of the hippocampus sits posterior, further back into the medial temporal lobe. Welcome to this tutorial on the neurobiology of emotion. I'm speaking to you from the home of some friends of ours here on Lake Gaston near the border of the state of Virginia and the state of North Carolina. And being in such a lovely setting as this certainly brings to mind lots of emotions. And in this tutorial, we get to explore together what's going on in our brains when we experience a beautiful setting like this or some other kind of emotion. And I'll be speaking to you about some examples of some of the darker sides of our emotional experience. But let's have a balanced perspective and also consider those wonderful moments in life when we get to enjoy a beautiful lakeside experience such as this, and consider the brain basis of emotional processing. So I'll see you back in my own home shortly, and we'll have a chance to dive deeply into this topic then. Welcome to what I think will be a really fascinating tutorial on the neurobiology of emotion. This topic pertains to several of our core concepts in neuroscience. Of course, we are once again exploring the complexity of the human brain, but we will also be talking about a kind of intelligence. It's been relatively popular in recent years to talk about emotional intelligence, to recognize that within the domain of affective experience, there is a certain kind of intelligence or a certain kind of expertise that seems to vary across people. I'm not really going to talk about that, but I do want to at least buy into the idea that there is a dimension of intelligence that pertains to our emotional experience and expression. And indeed, like all other dimensions of intelligence, these faculties arise as the brain reasons, plans, and solves problems. And then, lastly, I believe this core concept is certainly in view today as we talk about emotions and affective behaviors, and that is that the brain endows us with a natural curiosity to understand how the world works, including the world of our emotions and our human relations that are so critically dependent upon the means by which we can communicate using emotional cues as well as verbal cues. Well, my learning objectives for us today are several. I want you to be able to characterize emotion in neurobiological terms. That is, to characterize emotion as a form of associative learning. I want you to be able to understand something about the brain centers that appear to do this job of associative learning in the emotional domain. And two particular brain centers that we'll spend much of our time talking about today are the amygdala and the orbital and medial sector of the prefrontal cortex. So I want you to be able to understand how these structures participate in the experience and expression of emotion, and I'd like for you to be able to differentiate the role that each might play. And then lastly, I want you to be able to discuss the involvement of the limbic forebrain in decision making. And this will get us into really a fascinating discussion of really what is the relationship between emotion and reasoning. Are they really at odds the way we in the West and in the tradition of Western philosophy might have thought, or might they actually operate in some more synergistic way? Well, we will address that at the conclusion of the tutorial today. Well, let's begin by asking the primary question. What is emotion? Now, I've already talked some about how difficult it is to articulate the content of emotion using the conventional tools that are available to our explicit representational system, that is semantic symbols that can come to represent thoughts and ideas. Well, emotion seems to pertain more to implicit modes of processing rather than explicit. And perhaps this is why it is such a challenge to articulate in the same kind of detail with which we can, well, let's say, describe the sights and sounds of the bird who's singing up in the trees behind me. But this is a glorious problem that we face as human beings, is it not? Perhaps this is where artistic expression comes from. Where music, where forms of literature such as poetry, where the visual arts, the performing arts might derive their principal inspiration from this challenge of conveying a sense of emotion without necessarily having at our command the tools by which we typically ascribe meaning to explicit content. Well, more on that as we go along here. But let's appeal to some thoughts from really one of the founding fathers of biological psychiatry, William James. What did James have to say about emotion? Well, more than a century ago now, James described it this way. He said, if we fancy some strong emotion and then try to abstract from our consciousness of it all the feelings of its bodily symptoms, we find that we have nothing left behind. No mind stuff out of which the emotion can be constituted. Now, I'm not necessarily promoting all of the ideas and concepts put forth by William James more than a century ago, but I certainly think he was onto something here by suggesting that if we want to understand emotion, we need to understand how the brain moves the actions and interactions of the internal bodily state. James went on to say that, I say, for us, emotion dissociated from all bodily feeling is inconceivable. So James was quite intrigued with the relationship between body and brain, between mind and body, and how that interaction might give rise to emotional experience. Well, James and at about the same time, Lang together came up with pretty much the same concept. And together, this is sometimes called the James-Lang theory of emotion. It's one of the very earliest neurobiological theories of emotion. And here's how this worked. Well, William James imagined that the trigger for an emotion would be some kind of stimulus that would activate our peripheral receptor system. So this could be, let's say, the sound of footsteps in a dark alley rapidly approaching from behind. It could be an alarming somatic sensory stimulus. It could be a visual stimulus, or the combination of all of that. So this would be the proximal trigger of the emotion. But we're not yet there in expressing that emotion. So what James had in mind was that this emotion-provoking stimulus would be processed through our sensory systems, and this would lead to something like a reflexive activation of our motor system, both our somatic motor system and our visceral motor system. So smooth and striated muscle activities would be engaged. That would lead to yet a secondary sensory event where peripheral responses of these effector systems would be detected and sent back on into our relevant sensory divisions of the brain, principally the somatic sensory cortex and our visceral sensory cortex in the insular regions. These would be integrated, and it would be the integrated perception of this secondary sensory feedback that triggers the emotion. So in James's concept, the emotion then is elicited by peripheral feedback. This is why he so famously stated that we are afraid because we tremble. So note the causal direction there, that the fear is the consequence of the physical act of trembling. Well, these ideas were controversial right from the start. There were critics at the time who challenged this idea, and there are critics today who continue to find fault with this notion. William James emphasized the importance of smooth and striated muscle effector systems, not just in the expression of the emotion, but in the actual experience of the emotion itself. Remember, it's the secondary sensations derived from these effector systems that elicit the emotion in the James-Lang theory. Well, one source of criticism in the early days of understanding the physiology of the central nervous system was, well, if this is the case, then people with spinal cord injury ought to be severely impaired in their emotional experience, and that didn't seem to be the case. And more contemporary critics continue to basically attack this idea from the same perspective, and that is by challenging the postulated obligatory interplay between the body and the peripheral structures. Well, I think these criticisms are well-founded, but potentially underestimate the potential of the brain to represent bodily action, and to do so in a vicarious way that perhaps makes this relationship between body and brain in the production of emotion non-obligatory. We'll come back and talk more about this as we explore a more contemporary notion that is built upon this James-Lang theory of emotion. Well, I want to give you a neurobiological framework for approaching an understanding of emotion. And this framework will put us in a position to actually do experiments and test hypotheses that might begin to get at the neural mechanisms of emotion. And this framework is essentially proposing that we consider emotion as a form of associative learning. So here's how this might go. We imagine that emotions would result from the association of sensory stimuli with primary reinforcers. So the sensory stimuli, they might be interoceptive, that is, they might be derived from within the body pertaining to, let's say, visceral sensory signals derived from our gut and from other viscera of the body. They may be exteroceptive, derived from our interactions with the environment outside and be processed through our special sensory systems. The primary reinforcers are those experiences that have some intrinsic value to them. They may be rewards, which could be anything for which we might work, such as a pleasant taste or a tactile sensation. These reinforcers could be punishers, which would be anything that we will actually work to escape or to avoid. It could be an aversive taste. It could be a painful experience of one dimension or another. So the job then for building up an emotion would be to create some kind of association between primary reinforcers and sensory stimuli. And so here's how we imagine this might happen at the neuronal level. We believe that there are neurons in important structures in the brain. We'll spend a bit of time talking about the amygdala and the orbital medial prefrontal cortex. So we imagine that this might be a neuron in one such structure, such as the amygdala. So the amygdala in particular is well positioned to do this job of associative learning. It's receiving inputs from a variety of sensory systems. In fact, virtually all of the sensory systems provide more or less direct input into parts of the amygdala complex. Now, these sensory stimuli are in themselves neutral. They just might be sounds or sights or somatic sensory stimuli. But if those stimuli are present at the same time as a primary reinforcer, then we imagine that there may be some kind of associative learning that can take place. So again, the primary reinforcer could be that pleasant touch or taste, that aversive or painful stimulus. So we imagine via the convergence of very different kinds of inputs onto the same postsynaptic structures, we have the potential for synaptic plasticity to do the job of associative learning. Now, you'll recall the mechanisms of synaptic plasticity that we might have in mind here would include long-term potentiation, long-term depression, and in particular, if we're talking about building up an association of a strong stimulus with what is very likely initially a weak stimulus, then I hope you will recall the mechanisms that allow long-term potentiation to operate, and thereby increasing the impact of this sensory stimulus that was initially neutral and weak, but following associative learning might take on a life of its own due to the strengthening of the synaptic connection. Well, this might be a good time for a study question. So I'd like to remind you of what you learned way back in Unit 2 of the course, where we had a chance to consider in some depth the mechanisms of synaptic plasticity. So take just a minute and respond to this study question. All right, well, let's move on now. And we want to begin to dive into the relevant brain anatomy that is in a position to do this job of associative learning. And as we get into this anatomy, we really want to take on three kinds of topics as we explore the neurobiology of emotion. We want to talk about these effector systems that very well may be critical for the elicitation of the emotional response. So I will very briefly remind you of our parallel inputs to our effector systems that are positioned to do the job of motivating somatic motor and visceromotor activities in the experience and expression of emotion. Well, we then want to move on and spend a fair bit of time talking about the cognitive aspects of emotion, and this is where we begin to understand where the content of our emotion, the significance of that emotion, comes from. And then finally at the end, I want to spend just a few minutes proposing an idea that would help us understand the more subjective aspects of emotion. In fact, the most difficult of all aspects of emotion to explain in neurobiological terms, and that is the feelings associated with an emotion, One might have, for example, the somatic and visceral motor aspects of emotion on display. One might even have a sense of the significance of these emotions and what they contribute to cognition. But why should they feel the way that they do? So I think it's important to recognize that the feeling of the emotion is its own neurobiological construct, which might be separate from the somatic and visceral motor display associated with the emotion, and might be different from the role of that emotion in cognition. Okay, well, what kind of brain system might be involved with these various dimensions of emotional experience and expression? Well, over the course of the 20th century, studies of the emotions have led us into the ventral and medial parts of the forebrain, into what has been proposed to be called the limbic system. Now, I don't particularly favor this term limbic system, because once you use the word system, you're implying some kind of unimodal operation. And I think what we now know is that the set of structures that form a limbus or a rim around the medial part of the hemisphere really is not a unimodal system at all. In fact, there are multiple divisions of this classical limbic system that seem to be largely engaged in functions that can be dissociated. So I will refer you to a separate tutorial where I explore some of these topics in a little bit more detail, and I will actually show you some of the key centers in the brain anatomy lab, again to refresh your understanding of where these structures actually are in a human brain. But just to make this point, I do want to just go through some of the key anatomical centers that are associated classically with this limbic forebrain and highlight some of the distinctive functions that are associated with each. Well, believe it or not, the concept of the limbic system really began with Pierre-Paul Broca, the famous French physician and anatomist who initially proposed the idea of functional localization for the production of speech. So that same Broca who gave us Broca's area is the individual who coined the term limbic lobe, or le grand lobe limbique. Now, in Broca's day, what was noted was the strong connection between the olfactory bulb and this grand limbic lobe. So indeed, there is an olfactory division of this limbic forebrain that Broca recognized, and we know that this includes the olfactory bulb and the olfactory cortex. And the olfactory cortex extends over much of the ventral and medial distribution of the forebrain, including structures that are associated with this limbic forebrain. And so that olfactory cortex, together with the olfactory bulb, is involved in special sensory functions associated with olfaction, of course, with the sense of smell, and together with gustation, olfaction and gustation contribute to a sense of flavor. So there is indeed an olfactory division of the limbic forebrain. There is also what I like to call a parahippocampal division of the limbic forebrain. Now, the circuitry of the hippocampus was really a hot topic of investigation in the mid-20th century, and one of the leading anatomists and physiologists that advanced understanding of this part of the brain was James Papes. And some of you may be familiar with the concept of Papes circuit as being integral to the structure and function of a limbic system. Well, much of Papes circuit actually pertains to what I like to think of as this parahippocampal division of the limbic forebrain. It involves the hippocampus, related regions in the parahippocampal gyrus that we might call the hippocampal formation. There are connections that run through the anterior nucleus of the thalamus via the fornix and a secondary relay that then impact the posterior portion of the cingulate gyrus. There are also important connections, of course, that run through the posterior parts of the hypothalamus, including the mammillary body. So many of the elements of Papes circuit actually pertain to this parahippocampal division. And the functions of this parahippocampal division seem to be those that pertain to what I've been calling explicit processing. So this would include episodic memory acquisition and consolidation, and it would include spatial mapping. Not visual mapping, but mapping of our location within this broader environment. So these functions pertain to the explicit representation of the here and the now, understanding where we are in our environment and having the capacity to then take that knowledge and build it up into memory. So these are functions associated with this parahippocampal division. Well, lastly, there's a major division of the limbic forebrain that we'll spend the rest of our time talking about, and it's centered on the amygdala and the orbital and medial parts of the prefrontal cortex. So these two regions of the telencephalon are intimately interconnected. They also maintain connections involving the cingulate gyrus, the temporal pole cortex, and of course they have loops that run through the basal ganglia, through the ventral striatum, the ventral pallidum, as well as through the mediodorsal nucleus of the thalamus, which then projects back on into these cortical regions. The amygdala also has very extensive connections into the medial part of the hypothalamus, and that allows it to have access then to the central circuits that regulate our visceral motor system. The amygdala also has direct projections to the brainstem, where it can modulate brain systems that are responsible for organizing visceral motor output at that level of the nervous system, and also brain systems that can have an impact on our states of vigilance and attention. So this amygdala and orbital medial prefrontal division is involved with implicit processing. So it's coordinating visceral motor activity. It is going to be central for the experience and expression of emotion. It's responsible for producing appetitive drives, and it will have a major influence over the expressions of social behaviors. So what I want you to pick out here is really this key contrast between the idea of explicit processing, which is associated with the parahippocampal division, and implicit processing, which is associated with the amygdala and the orbital medial prefrontal cortex. And it's really this implicit processing that is important for understanding the neurobiology of emotion. Now, I want to next just briefly remind you of the parallel pathways that we have from the forebrain to our motor neuronal pools that are expressing somatic motor and visceral motor behavior. So, of course, we remember that for the expression of volitional movement, we have descending projections that are derived from the posterior part of the frontal lobe, where we have our motor cortex, projections that are mainly in the lateral part of the spinal cord pertaining to the expression of skill with our distal extremities. In order to set the stage for the expression of that skill, there are medial projections, many of which run through the brainstem reticular formation. And these medial projections adjust posture, set the gain of local reflexes that allow for the coordinated expression of skilled behavior. Now, I'll just remind you of the point that we made briefly in Unit 4 when we talked about the organization of the motor system, that there are indeed parallel projections that are derived from the limbic forebrain that are involved in the expression of specific emotional behaviors, as well as in setting the stage for the expression of those behaviors. And these are projections that are outside the classical pyramidal and extrapyramidal pathways. They run through rather diffuse projections that are still not well characterized or understood, but they can gain access to the same kinds of pools of autonomic preganglionic cells and even our somatic motor neuronal pools that are involved in coordinating the expression of somatic motor and visceral motor behavior. A great example of these parallel pathways concerns the organization of facial expressions. Remember, we talked some about how facile we are as human beings in telling the difference between a genuine emotional smile and what we might call a contrived or fake smile. Recall the last time you sat in front of a photographer and tried to produce a smile on command. What you were doing was activating your volitional motor systems, which pertains for control of the face to the lateral aspect of the precentral gyrus, where the command centers governing the movements of the lower part of the face are found. So that voluntary smile, that is the product of your volitional motor system, is activated via this posterior frontal cortex that is providing input to the columns of motor neurons in the facial motor nucleus that are driving the activities of the lower part of the face. So again, those are coordinated out here in the inferior segment of the precentral gyrus. Now, when we are actually engaged in a genuine emotion, that smile is coming from more medial parts of the forebrain that are gaining access to the lower motor neurons that are governing the expression of the face. But this system, this medial system, which is actually organized in a cingulate motor region, which is found in the banks of the cingulate sulcus, right about here is where we would find our cingulate motor area that's involved in generating emotional facial expressions. So there are very different pathways that are derived from different parts of the forebrain that converge in the facial motor nucleus and can sculpt the activity of facial muscles in a way that is conveying emotional content. And we're quite good as human observers in differentiating those patterns of facial muscle activity that are contrived from those that genuinely express true emotional content. Well, that's all that I intended to say regarding the expression of emotions via the motivation of our somatic and visceral motor effectors. Let's move on now and talk about how the content of emotion is integrated with other dimensions of cognition and can provide some significance to the context in which we are integrating our experience of the moment. So this really brings us to our two key brain systems for learning these associations that I've been describing between primary and secondary reinforcers. These two brain systems would be the amygdala and the orbital medial division of the prefrontal cortex. So let's take a closer look at the amygdala, consider its structure, and then some of its functional properties that pertain to emotional experience and expression. So as I've reminded you in a recent tutorial, the amygdala is a massive gray matter that's found in the anterior and medial part of the temporal lobe. Now, the amygdala is largely anterior to the hippocampus and the temporal horn of the lateral ventricle, but there is a little bit of a region of the posterior amygdala that overlaps with the hippocampus. But for the most part, think of the amygdala as anterior to the hippocampus in the medial part of the temporal lobe. Now, as we look at the amygdala, we see that it is a true complex. It's not just one mass of cells, but there are many subdivisions to this region. There is a cortical region to the amygdala, and this is in fact the cortex that we see protruding into that structure called the uncus on the medial aspect of the parahippocampal gyrus. There we find a collection of subdivisions that we call the medial group. Now, just lateral to that medial group, in a region that we sometimes call the temporal stem, it's a region of the brain where the temporal lobe attaches to the posterior frontal lobe in the overlying insular region. This is where we find the central group. So the central group of nuclei is quite important in providing the output from the amygdala into the medial part of the hypothalamus. But by far the largest division of the amygdala in the human brain is this basolateral group of nuclei. So we could further subdivide this, but for our purposes, we'll just consider this collectively as the basolateral amygdala. This basolateral group of nuclei in the amygdala maintains extensive connections with the orbital and medial part of the prefrontal cortex. So there are strong bidirectional interactions that happen between these two telencephalic regions that are so important in the experience and expression of emotion. But there are also less direct projections between the amygdala and this orbital and medial prefrontal cortex that run through the basal ganglia. So the amygdala is a very prominent source of inputs to the nucleus accumbens and other components of this ventral striatum. The ventral striatum in turn projects to ventral pallidum, substantia nigra pars reticulata, and from there those pallidal structures send outputs to the medial dorsal nucleus of the thalamus. So this is the specific thalamic nucleus that then gives rise to projections up to this orbital medial prefrontal cortex. So there's something of a triangular relationship here between the amygdala, the mediodorsal thalamus, and then the orbital and medial prefrontal cortex. And these structures can interact in various ways that we suspect are quite important for triggering emotional experience and expression. And let's not forget about the role of the basal ganglia in all of this. The basal ganglia is likely involved in producing a shift in the experience of emotion, and this might be consistent with the processing of a reward signal or a signal that predicts the experience of reward. Now, the centrality of the amygdala for learning the associations that produce emotion has been made much more clear from the use of animal models that have studied one particular dimension of emotional experience, and that is the emotion of fear. So, in laboratory studies of fear conditioning, we've come to understand how the amygdala is really the key site for associative learning. So here's how these experiments typically go. So there is a innocuous tone presented to a laboratory animal, and that tone is really neither particularly pleasant nor aversive to the animal, we imagine. It's just a neutral sensory stimulus. Now, that auditory stimulus is going to be processed, of course, by our auditory pathways, which lead into the auditory thalamus, the medial geniculate nucleus. The medial geniculate nucleus then projects to the auditory cortex, which relays signals into the amygdala. Now, interestingly, this medial geniculate nucleus also has direct access to the amygdala. So we imagine that while the cortical relay provides a more highly processed, more integrated sense of that stimulus, this direct projection from the thalamus to the amygdala is more of a very fast, kind of crude signal that is indicating the presence of that stimulus, not necessarily details about the tonal structure of that stimulus, but rather simply just the presence of this stimulus. Now, there are other projections into the amygdala from all other sensory systems, including those that would detect the presence of a noxious stimulus. Now imagine if that auditory tone is paired with something that the animal finds unpleasant, perhaps a brush to the underside of the animal through the floor of a cage, for example, or maybe a mild electrical shock applied to the floor of the caging. Well, this is a kind of aversive stimulus that we might consider to be a primary reinforcer, in this case a punisher. Well, the amygdala is going to be receiving input from these other sensory pathways, somatic sensory pathways in this case. So we imagine that this is going to be a very strong signal into the amygdala that might be initially paired with a relatively weak signal. And the pairing of these two inputs in time and in space, that is within the dendritic arbors of these neurons of the amygdala, results in associative learning. And as a consequence, the auditory tone now grows in its strength and its impact on amygdala neurons. The output of these amygdala neurons then is going to impact somatic and visceral motor activity and produce behavior consistent with fear and anxiety. Now, after some period of pairing these two kinds of inputs, the auditory stimulus and the aversive tail brush or electrical shock to the feet, this is going to produce the behavior associated with fear in this animal with the sole presentation of the auditory tone. So this is really one of the chief cellular examples that we have of emotional associative learning. And we know that something like this is taking place because we can impede this process by blocking NMDA glutamate receptors, which is suggesting that something like long-term potentiation is mediating this associative learning here among these amygdaloid neurons. We also know much more about the detailed circuitry that's involved in mediating the expression of this fearful behavior and how that's processed through intrinsic networks within the amygdala and then eventually via the output of the central group of nuclei to the medial hypothalamus and brainstem. Well, I think that this has been a wonderfully productive model, but some have questioned its relevance to human emotion, especially human fear. But I think that the relevance is real and compelling, in large measure because of studies of one particularly instructive patient who has come to the medical literature in the last couple of decades and has been studied extensively with a group based at the University of Iowa here in the United States. And this group has done just a heroic set of studies of this particular patient in characterizing her impairments in her emotional life, but also in her everyday real life experience in the real world. Well, this is a patient that is known in the literature as patient SM or patient SM46. Patient SM has been diagnosed with a condition that has led to the bilateral calcification and atrophy of her anterior and medial temporal lobe. And in particular, it's led to the bilateral destruction of much of her amygdaloid complex. However, this condition has spared the surrounding neocortical regions and, importantly, has spared the hippocampus. So patient SM has really provided a rare opportunity to explore the impact of a selective lesion of the amygdala with sparing of surrounding portions of the medial temporal lobe. What was quite obvious in the initial evaluations of this patient is that she showed no primary sensory or motor deficits, and she performed quite well on what was at the time the standard battery of neuropsychological tests. Yet she had real problems in her daily life, and the challenge to the investigators was to come up with a better means of testing her functions in the clinical setting that would resemble real-life experience. So they decided that they would really focus on the capacity of this patient to read emotion in the human face. And so they did a series of studies that were reasonably well controlled. They compared her performance to the performance of a group of patients that had injuries to their brains, but outside of this medial region of the temporal lobe. And what they did was they asked patients to rate the content of various primary emotions, such as surprise and sadness and anger and disgust and fear, in the presentation of portrait views of the human face. And what we're seeing here in this plot is simply a way of quantifying the ratings of these various primary emotions that these subjects recognize across this inventory of photos. And what you see in the bold orange line is the ratings of fear that were present in this inventory of human faces. Now, when patient SM was asked to do the same, these were her ratings. So you can see that she did quite well on ratings of, let's say, anger and disgust and happiness. Her ratings of sadness were likewise pretty high and relatively normal. For some reason, her surprise was somewhat down. But clearly, the one emotion that she really struggled to rate and recognize in the human face was fear. So in this bold orange line, that's her rating of fear in the same inventory of human faces. Now, these investigators went on to study various dimensions of patient SM's life, and fairly recently, they utilized mobile technology, and they asked her to respond to a short survey that asked her to reflect and introspect and rate her experience of various emotions at different times of day over about a 3-month period. And what we're looking at here are her responses to these various prompts about her ratings of her emotional experience. And the scale here is a percent of the maximum possible performance that she might have displayed across these various dimensions of emotion. And what's very clear is that she hardly ever at all self-reported experiences of emotions that we might consider associated with fear. Now, there's really a fascinating set of papers that I would encourage you to read if you're interested in learning more about patient SM. In short, she appears to be an individual that is impaired in her ability to read fear in the human face. And consequently, she appears to be severely impaired in the actual experience of fear in day-to-day life. I think what we've learned from patient SM is that in the human brain, the amygdala is an important center for the experience and expression of emotion, especially the emotion of fear. Well, why might this be helpful in everyday life? Well, I think we're learning more about the basis of social cognition and how it relates to brain systems. And what's becoming clear is that the amygdala and its role in our emotions is very important in mediating various aspects of social cognition. One important aspect of social cognition that's relevant is our evaluations of the trustworthiness of another human being. So what's being shown here in a very clever set of experiments is the activity of the amygdala when human subjects are evaluating human faces. And the evaluation instructions are either explicit about making a determination about trustworthiness, or the evaluation is implicit. That is, the participants in the study are directed to make some other differentiation, such as whether the faces are of university students or secondary education students. But in both cases, what we see is a clear relationship between activation in the amygdala, left and right, and judgments of a person's face being untrustworthy. So what's very clear in these data is that when amygdala activity is high, our judgments of trustworthiness are low. That is to say, if we are in a social situation and interacting with an individual and we're beginning to believe that we might not want to trust this person, that sense of not wanting to trust seems to be associated with elevated levels of amygdala activity. Now, I think it's fair enough to think of the amygdala as our brain's early warning system, that it's providing us with a sense that something just might not be right. It's not necessarily signaling anxiety or even fear, but rather it's increasing vigilance so that we can attend to the rich array of sensory stimuli that are defining the nature of this interaction that we might be having. So let me try to summarize what I've said so far about the amygdala. So the amygdala clearly plays a primary role in the association of sensory stimuli with reward and punishment. But I think more generally, the amygdala has an important role to play in cognition. The amygdala modulates information processing in the forebrain, and it has an impact on our perceptual systems. It can enhance the awareness or the attentiveness of our perceptual systems to aversive or arousing stimuli. The amygdala can have an impact on memory, on our mnemonic systems. It can enhance memory for emotionally charged events. Now, this provides us with a framework for thinking about what might happen when there's not enough signaling coming out of the amygdala, or perhaps when there's too much. So with insufficient amygdala activation, or amygdala hypofunction, we can expect there to be deficits in the efficacy of information processing. Perhaps this helps us understand patient SM. So the sensory signals are being processed, but she's simply not attending to them in a way that would impact her judgments about potential threat. So we imagine in patients like SM, the early warning system is defective. It's not going off. It's not signaling the need to have a heightened sense of vigilance, to have directed attention about the sensory stimuli that might be very significant in giving us clues about the nature of the social interaction with which we are engaged. As a consequence, patient SM and perhaps others like her have been the subject of violence, of domestic abuse. So I can imagine that living without the brain's early warning system might put one at risk in a variety of social circumstances where the intentions of other people are not always so clear, and it requires reading these emotionally nuanced signals in order to inform our judgments of trustworthiness. Now, there are also problems with amygdala hyperfunction, that is, when there are too many signals coming out of the amygdala. Imagine what it might be like to be living with an overactive early warning system. As a consequence, we might expect there to be excessive vigilance, perhaps even anxiety, or perhaps even overt fear. So let's turn our attention now from the amygdala here in the anterior and medial part of the temporal lobe to the orbital and medial part of the prefrontal cortex. So I'll just remind you again by showing you in this brain model, the orbital cortex is the ventral portion of the frontal lobe that sits just above the orbits of the eyes, and it continues on to the medial surface of the hemisphere, where we find just above this orbital cortex this medial prefrontal sector involving the medial portion of the superior frontal gyrus, as well as the anterior part of the cingulate gyrus. And these areas continue and reach an interesting region called the subcallosal area, right below the genu of the corpus callosum. So we've learned quite a bit about the functions of the orbital and medial part of the prefrontal cortex from studying their anatomical connections. We know that this part of the prefrontal cortex receives inputs from the amygdala, as I've already described for you. It's also receiving inputs from virtually all of the other sensory systems. And it's also receiving input from the associational cortical areas, especially the associational cortex of the parietal lobe and the temporal lobe. We think that this cortex is involved in emotional learning. I'll say much more about that in just a few minutes, as well as in the interpretation of social cues. This cortex is involved in planning appropriate social behavior, the formation of advantageous real-life decisions. We might consider this to be a contribution to our faculties that are involved in reason and rational decision-making. So just in a little bit more detail now, the orbital medial prefrontal cortex is involved in emotional processing, and it does so in a way that allows our emotions to provide a source of input that will help us to interpret social cues. And as a consequence, it will assist us in planning appropriate behavioral responses as we engage in social interactions. Now, the role that this portion of the prefrontal cortex plays in emotion bears much similarity to the role that we ascribe to the amygdala. It's involved in emotional learning, but especially when the rewards are in the form of olfactory signals and gustatory signals, that is, in the form of sense and in the form of food rewards. But it's much more than that. The orbital and medial prefrontal cortex is involved in the ongoing analysis and modification of behaviors, especially when the reinforcement contingencies are rapidly changing. We consider this a form of emotional relearning. Imagine just the complexity of being, let's say, in a mixed company in a social situation, be it at a club or at a party or just engaged with a group of friends and perhaps some new acquaintances. There are many complex dynamics at work here, and sometimes the kind of conversational devices that work in one interaction with one kind of person may have a very different meaning with a different type of person, perhaps a person just of a different background, maybe a different gender. But the point is that these social situations are highly dynamic and require a great amount of agility. And it's that social agility that is a particular challenge to this orbital and medial sector of the prefrontal cortex, and the role that it plays in interpreting these kinds of complex social cues to guide appropriate behavior. We think that this sector of the prefrontal cortex is necessary for the assessment of future consequences and the implementation of advantageous decisions. And I'll have more to say about that in just a few minutes. Now let's consider what might happen with injury to this part of the brain. And much of what we've learned about such cases stems from studies of that famous case of Phineas Gage back in the 19th century, who had this iron rod blown through the anterior part of his cranium with severe damage to his orbital and medial prefrontal cortex. Well, in the decades since the life and times of Phineas Gage, other patients have been identified, and now there is a cohort of patients that are being followed who have had damage from either the growth of tumors or neurosurgical resections or strokes or traumatic brain injuries that have shed considerable light onto the contributions of this part of the brain to human cognition and social behavior. So what we've learned is that people with injury to this part of the brain show typically no impairments in sensory function and in volitional motor control, except for impairments in the olfactory sense and in the gustatory sense. And together, those combine to give us a sense of flavor. So these dimensions of the chemical senses are impacted with damage to this part of the brain. But otherwise, the rest of our sensory systems are intact, and for the most part, our volitional motor system is intact. Again, with our standard means for doing neuropsychological assessments, these patients seem to be doing perfectly fine. Yet there are real impairments in emotional experience and expression. And perhaps as a consequence, there are impairments in the capacity of these individuals to make rational decisions, especially when these decisions pertain to the domain of personal and social affairs. One very curious feature of this impairment in decision-making is that these patients tend to have a pathological inability to make advantageous decisions when it comes to real-life situations. As we'll see, it's not just that they behave at chance. It's that they actually tend to make poor decisions that can have serious consequences for their real-life circumstances. Now, much of the leading work that has gone on in the study of patients with damage to this part of the brain are in fact the same group that has brought to the attention the case of patient SM, a group in Iowa that has studied these patients with brain injuries. And this group quite presciently recognized that the standard battery of neuropsychological assessments really did not seem to be sufficient to get at the true nature of the real-life deficits in these patients. So they proposed a new kind of test that they called the Iowa Gambling Test. So this test involved a set of playing cards that could be divided into 4 decks, and the patient is instructed to draw a card from one of these decks. What the patient doesn't know is that there is a certain protocol for providing financial reward or financial penalty for drawing a card from one of these 4 decks. And the way this is set up is that the patient's given a certain allotment of money at the beginning of the experiment, and the patient begins to choose from these decks of cards. And 2 of these decks of cards we might call disadvantageous, because while there may be large gains, there are more frequent moderate losses or infrequent large losses. And the aggregate result of drawing cards from these particular decks over the course of this experiment is that there will be net financial losses if these patients choose from decks A or B. However, if the patients are drawing cards from decks C or D, there may be small monetary gains, but the losses are either going to be small themselves or they'll be much more infrequent and of a more moderate dimension. So this is a more conservative strategy that leads to net financial gain. And over the outcome of the experiment, we might consider this to be an advantageous decision-making strategy. So what we find in normal subjects over the course of time is that most of us very quickly begin to explore the disadvantageous decks, and over time we choose less and less from the disadvantageous decks and more from the advantageous decks. So we end up with a net financial benefit. That is, we end up making a little bit of a profit as we explore the rules that govern the financial gains and losses in this game. Now, interestingly, what patients with damage to their orbital medial prefrontal cortex do, and also patients with amygdala damage, they tend to choose preferentially from this disadvantageous deck, and it never quite seems to go away. They will choose from the advantageous decks as they begin to figure out that that might be a useful strategy, but they never quite seem to do so with greater prevalence over choosing from the decks that lead to the financial loss. It's as if what they are drawn to is the impulsive prediction of significant reward, even though over time that strategy will lead to a net financial loss. And this paradigm in what is called this Iowa gambling task is getting a little bit closer to the real-life impairments that these patients experience as they make decisions in the real world, many of which are financial decisions. So perhaps not surprisingly, many of these patients end up going bankrupt because they terribly mismanage their resources. They also suffer from broken human relations within their family and within their extended circle of friends. Again, because their decisions are based mainly on assessments of short-term gain. They seem to fail in the proposition of projecting out long-term consequences of their action. And that's where the connection between emotion and reason seems to come into play. Now, the lead investigator that has developed this line of thinking is Antonio Damasio, but many others of his colleagues have contributed over the years to the study of these patients and to the formulation of the ideas. And I, for one, am quite compelled by these ideas and think that Dr. Damasio and his team might actually be onto something quite important. So Damasio and colleagues have proposed what they call the somatic marker hypothesis of reason. And as you'll see, this is very much a neo-Jamesian idea, reflecting William James and Lang's theory of emotion. So at the heart of this idea is the notion that rational decision-making entails a set of covert evaluations of future consequences. Now, these covert evaluations take the form of what Damasio calls mental images. And these mental images represent the possible outcomes of our decisions that might trigger states of body and brain. And Damasio calls these somatic states. So these are somatic states that would mark these mental images with emotional valence. Now, what he's getting at is that the brain might actually be representing the state of the body as it experiences the consequences of our decisions, and that these brain representations of somatic state then become a source of bias that can influence the outcome of these covert deliberations. Now, these somatic states may be truly somatic, and this would be very much a James Lang proposition. This would involve the activity of autonomic and skeletal muscular effector systems. But, as Damasio has argued, these somatic states might be vicarious. And what he means by that is that they may only involve the brain constructs, the neural representations of this visceral motor and somatic motor activity. We imagine that this would be focused in our parietal and our insular regions of the cerebral cortex. And here's where there's a bit of a departure with the classical James Lang theory. Damasio would propose that indeed the relationship between body and brain may not be obligatory for the operation of these mental images. Rather, in the parietal and the insular cortices, there may be a stored memory of body state that can be mobilized and used to mark that mental image with an emotional valence that reflects the way the body feels when experiencing one possible emotional outcome or another. So in Damasio's conception of decision-making, these somatic markers are biasing the covert deliberations towards the implementation of advantageous decisions. And this helps us understand what might happen with damage to these parts of the brain, where this important somatic marker function is disabled. As a consequence, without being able to bias our decision making towards future advantage, what we're left with is the allure of short-term gain, and we tend to act more impulsively without these somatic states biasing our decision making and our future orientation. Well, I'd like to conclude our tutorial by talking about this much more elusive aspect of the neurobiology of emotion. That is, the subjective feelings that are associated with our emotions. So where do these feelings come from? And why should they exist in the first place? As I suggested earlier, having an appropriate fear response need not actually make you feel afraid, but yet it does. So this has led to the proposition that perhaps feelings are the consequence of having emotions within a cognitive faculty that is capable of self-reflection, of introspection, of working memory. So the notion here is that perhaps feelings are a kind of emotional working memory. So imagine then that there are the appropriate stimuli that might trigger the experience of an emotion. As we've been discussing, the stimuli will impact our amygdala-dependent associative learning systems, as well as those associative learning networks in the orbital and medial prefrontal cortex. Of course, we will also generate explicit representation of these events through our hippocampal dependent memory systems. And together, the implicit and the explicit will interact in interesting ways between amygdala systems and hippocampal systems, but also they will influence our prefrontal networks. And it's there in the prefrontal cortex that we will have an immediate conscious experience of our emotional feelings. So you'll recall that when we talked about working memory in the dorsolateral portion of the prefrontal cortex, we described the activity of neurons that are maintaining representation of the goal that is motivating our behavior. Well, we think that something like that might be going on in the orbital and the medial sectors of the prefrontal cortex, where the object of implicit representation is being sustained in the activity patterns of this part of the brain. And from that perspective, perhaps the feeling associated with the emotion is a reflection of a kind of working memory. What we may be doing is maintaining in our immediate conscious experience the implicit processing that is motivating and guiding emotional behavior. So it's really this capacity for working memory, or there is this capacity for introspection that is giving us the feeling of the emotion. Now, perhaps you're wondering whether non-humans experience emotions in the same way that we do. Well, if you're curious about that, you might start with a fabulous monograph written by none other than Charles Darwin. And this has become one of the seminal works regarding the expression of emotion in not only humans, but in non-human animals as well. I think it's fair enough to state that the capacity for emotion is associated with the development of this orbital and medial portion of the prefrontal cortex, with this capacity for an emotional working memory being critical for the experience of emotion with the feelings that attend these states of body and brain. Now, when we survey the anatomy of our own orbital and medial prefrontal cortex, obviously we're impressed with the development of these regions of the human brain, and it's tempting to therefore conclude that there must be some dimensions of emotional experience that are uniquely human. Well, I don't think we know that for certain, but nevertheless, there is no missing the conclusion that our emotional experience is a well-developed capacity that is attributable to the development of this portion of the human brain. And the ability to experience and to express emotion is critical for social cognition and healthy human relations. Well, let's conclude this tutorial by leaving you with a few points of summary. So what I've tried to describe for you is that human emotion entails a wide range of brain and body states that are characterized by expressive somatic motor and visceral motor behaviors, by stereotypical physiological responses of the body, and by distinct subjective experiences. These dynamic states of body and brain are likely to arise as a result of an associative learning process that attaches emotional valence to sensory stimuli. Associative emotional learning involves two key centers in the forebrain: the amygdala in the anterior medial temporal lobe and the orbital and medial sector of the prefrontal cortex. The amygdala is a primary neural center for associative learning, and one important contribution of the amygdala is to process those signals that will increase our vigilance because they predict the presence of risk or threat. Another important neural center, of course, is the orbital and medial sector of the prefrontal cortex. This also contributes to emotional learning, especially when the contingencies of reinforcement are rapidly changing. And this is certainly one way to understand the complex dynamics of real life, real world social engagement. Well, I hope you've become as fascinated as I have in these parts of the human brain. And we are just now entering what I believe will be a great new era of discovery, where the mysteries of this orbital and medial sector of the prefrontal cortex and its contributions to cognition will be much better understood. And as a consequence, I think we will have much more insight into who we are as human beings. In today's tutorial, which is, as you know, the concluding tutorial of Unit 6 on complex brain functions, and indeed the concluding tutorial of medical neuroscience, I've chosen to wrap up the course with a topic that is of tremendous importance for anyone entering the world of healthcare because of the impact of addiction on societies all over the world, and indeed because of the risk of addiction for workers in the healthcare sector of our global economies, where opportunity and risk abound. So I want us to just think together very briefly about some of the mechanisms of addiction that pertain to the structure and function of the limbic forebrain. And to do so, we're going to back up just a little bit and talk briefly about the basal ganglia and the brain's intrinsic system for reward. So today's session pertains to several of our core concepts in the field of neuroscience. We will again talk about the complexity of the brain and illustrate the fact that the brain is indeed the body's most complex organ. We'll be talking about genetically determined systems and circuits that provide the foundation for the function and operation of the central nervous system, and in this case we'll be focusing on the brain system for reward. And then lastly, the phenomenon of addiction is a sad but compelling demonstration of how life experiences can change the nervous system. And the challenge for medical science and for rehabilitation professionals is to understand best how do these intrinsic systems of reward operate in the brain, and how might we best approach the process of undoing the grip of addiction that can come upon the brain's intrinsic reward system. So my learning objectives for you in this tutorial are first to be able to discuss the brain's reward system, and next to discuss the involvement of the limbic forebrain in mediating addictive behavior. Now, as we get into this topic, it will be clear that one very important brain system that is germane for understanding addiction is, in fact, a component of a brain system that we've already spent a fair bit of time wrestling with, and that is the basal ganglia. So I'll remind you that there are multiple streams or loops, as we sometimes call them, that run through the basal ganglia. We talked about a dorsal motor stream or a motor loop that principally runs through the dorsal parts of the basal ganglia. That is the putamen, which is a major component of the striatum, lateral to the internal capsule. The putamen has important connections to the globus pallidus, which then projects to the motor thalamus that modulates the expression of movement, specifically the initiation of movement or the suppression of movement. We talked briefly about a cognitive stream that runs through the caudate division of the striatum, the caudate nucleus. And this caudate nucleus has projections into the pars reticulata component of the substantia nigra. That's a palatal region that serves the anterior part of the head of the caudate nucleus, and it's involved in this cognitive loop that involves movements of the eyes as well as movements of the mind. As I've mentioned several times, what we look at is often what we are thinking about. and that can become the object of our plans for future actions. Now, in this tutorial, we're going to focus on the third major division of the striatum, the nucleus accumbens, which is an important component of what we've called the ventral striatum. And this ventral striatum has the same kind of circuitry that we've seen in the other divisions, that is a direct and an indirect pathway, but it's operating upon inputs that are coming from different parts of the telencephalon. And the output from the palatal component of this striatal loop is directed towards a non-motor thalamic nucleus, namely the medial dorsal nucleus of the thalamus, which then projects into our prefrontal cortical networks. So let's take a closer look at this ventral limbic loop in the basal ganglia, and that will help us understand a little bit more about how our reward system might be operating in the human brain. So as we see, this limbic loop really begins with inputs that are derived from regions of the telencephalon, such as the amygdala, the hippocampus, the orbital frontal portion of the prefrontal cortex, the cingulate gyrus, and even the cortex at the anterior pole of the temporal lobe. These are all important integrative associational areas that are involved in the experience of emotion and the integration of emotional experience with other dimensions of cognition. So these are the sources of telencephalic input into the ventral striatum. And specifically what we're talking about here as the major component of the ventral striatum is the nucleus accumbens. You'll recall that this is the division of the striatum that is continuous with the caudate nucleus dorsally and medially, and with the putamen laterally and dorsally. And this nucleus accumbens wraps right underneath the white matter of the internal capsule. So here's the internal capsule, and we see that on either side of the internal capsule are components of the striatum, caudate medially, putamen laterally, and then inferior to this internal capsule is where we find this nucleus accumbens. Now, the nucleus accumbens, the ventral striatum, then sends inhibitory projections into the pallidum. And the pallidum that we have in mind with our ventral limbic loop is called the ventral pallidum, as well as the reticulata component of the substantia nigra. So the ventral pallidum is a region just behind this section. We can't see it in this view, but it's a region posterior to the nucleus accumbens, but inferior to the globus pallidus. So it's a much looser, organized, reticulated network of pallidal neurons. Well, the output from this ventral pallidum and this particular component of the pars reticulata is directed at that medial dorsal nucleus of the thalamus, which is the principal thalamic nucleus that projects out to the prefrontal cortex. So the projections here are indeed directed back towards many of the same cortical regions that gave rise to input to this nucleus accumbens in the first place. Most importantly, the orbital and medial part of the prefrontal cortex. Now, what we've illustrated here is the direct pathway through the ventral limbic loop, and I hope you've recognized that based on the presence of these two inhibitory connections within the basal ganglia circuitry that, when active, serve the purpose of disinhibiting the thalamic projection to the cortex, and that disinhibition then provides a kind of a go signal for the engagement of the cortical circuits that are responsible for some kind of shift in behavior. So exactly what kind of behavior we have in mind is going to depend upon what is actually being integrated in this cortical network at the time, but we can imagine that it has something to do with affect, with emotion, with mood, perhaps even the kind of shift in affect that we would associate with the presence or the prediction of a reward. That is, we know something good's about to happen, and there's some kind of a shift in our emotional set. And it's that shift played out in this circuitry of the orbital medial prefrontal cortex that we think is really the instantiation of a rewarding experience. Now, I'm talking about reward. We imagine that there is some kind of operation like this that operates for the instantiation of the negative valence that we might associate with this kind of behavior, that is, with punishment, something that has a negative reinforcement property. Also involves a shift of some sort. So we imagine there's likely to be a different kind of network, probably also in the orbital and medial prefrontal cortex, but maybe in a slightly different location, that is responsible for initiating that shift towards the negative valence of punishment or deterrence, or this concept that, okay, this is a bad outcome, so I need to learn my lesson here and try to avoid this kind of contingency in the future. So hopefully this gives you a sense of how the basal ganglia might actually be key in understanding how the reward system of the brain might work, to either reward with a positive reinforcer or a negative punisher. Now, there's one critical component of this circuitry that I need to highlight before we move on, and that is found right here on the medial edge of the substantia nigra in the midbrain. This is the ventral tegmental area, which is critically important in modulating the way the circuitry through the ventral loop of the basal ganglia operate. So the ventral tegmental area is the source of dopamine for the ventral part of the striatum. So there are projections from this midbrain region into the nucleus accumbens where dopamine is released. And there are a variety of dopamine receptors. We've tried to keep it simple and only talk primarily about two kinds of dopamine receptors. In the striatum, we have D1 and D2 receptors. The D1 receptors are associated with the direct pathway. So in the illustration that we have here to the left, we imagine that the D1 receptors are being activated when ventral tegmental neurons begin to fire and release dopamine, affecting this direct pathway from ventral striatum to ventral pallidum, substantia nigra pars reticulata, and then back to the mediodorsal nucleus of the thalamus. But there's also an indirect pathway, not illustrated here for simplicity, where dopamine is going to interact with D2 receptors and suppress the firing of those indirect pathway striatal neurons. And together, when dopamine is present, the D1 and the D2 receptors work synergistically in order to facilitate the activation of the thalamocortical connection. Okay, well, let's turn our attention now to the firing patterns of these ventral tegmental area neurons, and that will give us some insight into the nature of the reward signal that we have in the human brain. So what we're looking at here is a raster plot of recordings from the ventral tegmental area. And we're looking at action potentials in a paradigm where an unexpected reward is provided to a behaving animal. So the individual dots that we see down below are individual action potentials on multiple repeats of the same trial. And up above, we have what we call a post-stimulus time histogram, which shows us the cumulated firing of this neuron across all of these trials. Now, at the arrowhead down below, we have the introduction of a reward. Now, this is unexpected to the animal, so this is a pleasant surprise, if you will. So with that present surprise, we have a marked increase in the activity of this ventral tegmental area neuron. And so this neuron is now releasing dopamine in the striatum after the presentation of this unexpected reward. And so this is inducing a shift in the activity of the reward circuitry of the orbital and medial prefrontal cortex, which is facilitated by the activation of its thalamocortical connection. So the striatum, the pallidum, disinhibits then the mediodorsal nucleus, And there's some kind of change in the activity patterns in this orbital medial prefrontal network. And presumably, that change, we don't yet understand exactly how this works, but that change is read out in the animal's behavior as the presence of rewarding stimulus. Well, now let's see a different experiment where there's a conditioning stimulus that's present. So the conditioning stimulus is coming to predict the presence of a reward. And after enough trials to reinforce this contingency, what we find is that the firing of the ventral tegmental area neuron now shifts from the reward to the conditioned stimulus. So what this neuron is signaling then is not necessarily the presence of the reward, but rather the prediction of the reward, the anticipation of the reward. So we can imagine why this would be quite important in understanding what happens as people become addicted, be it to a drug, be it to gambling, be it to sex. Whatever the experience or the substance of abuse happens to be, increasingly over time, the firing of the ventral tegmental area neurons, the release of dopamine, and the activation of the reward circuit becomes associated with the conditioned stimulus. Not so much anymore the consumption or the experience of the substance of abuse or the experience of abuse. In fact, if we continue to play this basic paradigm out in time, but now withhold the presence of the reward, what we find is that the ventral tegmental area neuron will continue to fire, at least for some number of repeated blocks of this experiment, when the conditioned stimulus is present, but it actually has a suppression of firing at the time at which the expected reward would have been delivered. But in this experiment, it was withheld. So the firing of this neuron becomes increasingly shifted then from the presentation of the reward to the anticipation of the reward. Now, let's look at a little bit of the molecular biology of what's going on both in this ventral tegmental area neuron, but also in its target, in the medium spiny neuron of the ventral striatum. Now, normally, as you know, there is the integration of inputs in these medium spiny neurons, which are these large neurons here shown in blue. Here to the right, here's the nucleus accumbens medium spiny neuron. And on the left, we have a ventral tegmental projection neuron, which provides input to this medium spiny neuron in the form of dopamine. So here we see a nice release of dopamine that would be signaling either the presence or the prediction of a reward. Now, this medium spiny neuron is also integrating inputs that are derived from the cerebral cortex, as well as structures like the amygdala and the hippocampus that are providing input here. We haven't talked a lot about this, but the dopamine neurons in the midbrain are also getting input from widespread regions of the cerebral cortex and probably lots of other structures in the brainstem as well. Now, what's meant to be depicted here in this slide is really, I think, a remarkable feature of the neuropharmacology of the system, and that is at just about every dendrite that's relevant in this circuitry, there are receptors expressed for virtually all known drugs of abuse that human cultures have ever discovered. And indeed, some of these receptors are even found on the presynaptic terminals that provide input to these cells. So, for example, in the nucleus accumbens, we have receptors that can interact with substances like cocaine and amphetamine, which can act to increase the presence of dopamine in the synaptic cleft of these medium spiny neurons. We have receptors for phencyclidine, for opiates, for the cannabinoids, all expressed on the synaptic junctions of these neurons with their inputs from the telencephalon or from the dopamine inputs derived from the ventral tegmental area. And in the VTA itself, we have opportunity for substances of abuse to modify the way these VTA neurons either respond to local GABAergic interneurons or from their cortical projections that are releasing glutamate. So the upshot here is that the brain's intrinsic reward system is expressing receptors that can interact with a variety of exogenous compounds that we can consume and can co-opt the function of this reward circuitry. So let's look at what happens following addiction in this system. So the details here are a little bit beyond our scope, I think, so we won't have to focus on them, but I do want to give you a sense of the physiology of what's happening here. So when this system becomes addicted, what we find are molecular changes within the neurons of the ventral tegmental area and the nucleus accumbens. There's plasticity of synaptic connections. There's very likely structural change in the distribution of synaptic terminals and the structure and distribution of the dendritic arbors as well. But from a physiological standpoint, the key feature here of this addicted circuitry is that there is a reduction in the ongoing release of dopamine that is normally reflecting the nuances of everyday life. That is, those momentary pleasures that we all experience from pleasant social relations, perhaps a kind word or a gentle touch, or the sound of our favorite song that we might happen to listen to on the radio or in our music player. All of these nuances of daily life that give us some sense of pleasure and reward are associated with the release of dopamine in the circuitry. It's exactly these nuanced rewards of everyday living that become diminished in the addicted circuitry. What we find in the addicted brain is that the release of dopamine is increasingly tied to the prediction of consuming the substance of abuse or experiencing the situation that is intended to provide for that reward. But as we saw in the neurophysiological experiments, it seems as though the release of dopamine is increasingly tied to the conditioned stimuli, not the actual consumption of the abusive substance itself. So what we see then in this circuitry is that the release of dopamine, as well as the phasic stimulation of this circuitry that's coming from cortical systems, is increasingly tied to these conditioned stimuli. So I think this is providing great insight to understanding addiction and addictive behavior. Perhaps this is why the presence of, let's say, drug paraphernalia, or the presence of acquaintances and other individuals that might otherwise be associated with abusing drugs, can have such a powerful grip on the addicted individual, because these are conditioned stimuli that are predicting the consumption of a substance of abuse. And in the presence of these conditioned stimuli, we imagine that these ventral tegmental area neurons might start increasing their firing, releasing dopamine, and predicting the consumption of the rewarding stimulus. But if the brain of the addict is anything like the animal model data that I just showed you, we can expect that the actual consumption of that abusive substance is no longer producing a release of dopamine. If anything, there might be a withdrawal of dopamine that would happen at that time. So addiction is a serious problem. It's not necessarily an intractable problem, but for sure we need to develop better methods based on neuroscience discovery for the treatment of this human condition. Well, I hope this has given you some insight as to what might be going on in your own brain when you have a rewarding stimulus. And if you do believe that you are at risk for addiction, I would urge you to seek counsel, perhaps seek medical intervention if that's appropriate. But otherwise, I think it's important for us to understand how this part of our brain works. Certainly any of us that are in a privileged position that might have means and access, it's critical for our health, for our wellness, for the health of our families and our relations, that we be on guard and that we protect ourselves, even this aspect of our behavioral health, based on what we're learning about the brain and its intrinsic systems of reward. Well, thank you for your attention. I hope I wasn't a bit too heavy-handed with you on this topic. I'm used to talking to medical students about this particular system, and at least in the medical profession, the temptations are very real. And the abuse of substances among the medical profession is certainly a problem that we have here in the United States and in many other places in the world as well. So knowing more about how the brain deals with reward and how the brain can so easily be co-opted by substances and experiences of abuse gives us better information that will allow us, I trust, to develop better strategies for maintaining the health of our brains, the health of our bodies, and the health of our social relations. Well, I'll see you next time as I wrap up with you our experiences in medical neuroscience.